outbreak and a low-path AI outbreak that still has relevant lessons, where to find the expertise and experience to vaccinate and immunize poultry in their varied field settings, a proposal for private engagement, and a way to move forward. Next. Not working. Anyway, just go ahead and advance the slides. I don't have any animation, so it should be easy. Next slide. Okay, so I'm going to tell you a little story about California. We had an H6N2 low-path AI outbreak, and I think it's fair to talk about low-path AI when the difference is when they're highly transmissible. Very similar in my mind to high-path AI. So we had an outbreak from 2000 to 2005 that just spread and spread and spread. Chickens were vaccinated. At first, they used a very severely unmatched H6 virus, and that virus was given in a killed, it was a killed vaccine. This mismatch, of course, allowed the virus to continue to circulate. It left flocks that were susceptible to later infections, which did happen, and I'll talk about that in a minute. And immunity post-vaccination was not tracked at all, and there were no movement controls put on birds that were vaccinated. At first, only egg layers were impacted. We had no end game in place and no real stated goals for immunization. Vaccine, we didn't state the goal, but the goal was clearly to prevent disease and egg production issues. All right. Okay, yes, so we can quit now because the thing's working. All right, so the outbreak was localized to egg layers in Southern California. If you're not familiar with California, we have a mountain range in the middle of the state that divides Southern and Northern California. We have egg layers in both sides of that mountain range. There was a company, a broiler company, that owned a spent fowl processing plant in Southern California. That's where all the birds at the end of their life cycle went for processing. That broiler company looked at their spreadsheet one day and said, what do we need this for? So they sold it. And what happened then? Exactly. What happened then is all of those chickens came to Northern California, right to the heart of broiler company. We had no pre-movement testing in place. We had poorly immunized or non-immunized flocks. We had new antigenic variants emerging, but it didn't matter. It could have been the old antigenic variants. The birds weren't actually immunized at all. We think that the first case that brought that virus over the Tehachapi Mountains was actually the first infected flock. And if we had done the serology, we would have found out that only half of the flock had ever been infected. The other half was not infected. They'd had a drop in egg production and an increase in mortality, but of course they did. They were at the end of their life cycle, so nobody cared, right? We didn't notice the clinical signs. So a fully adapted chicken virus then made its way into broiler country. Oops. Well, that was good. Okay. So what happened in Southern California, the layers spread the virus to egg layers in Northern California. Because of course, in Northern California, we were using the same spent fowl crates to move chickens out in Northern California that we used in Southern California, the same trucks, nobody was washing. Then the broilers got infected and the turkeys. And once the broilers were infected, the layers to the turkeys and the broilers back to the layers. But once the broilers were infected, all bets were off. They spread to everything. So the poultry industry So once the virus was in broilers, all bets were off and everyone was engaged. The poultry industry introduced its own movement controls and monitoring programs. H6, non-reportable, non-regulatory. So in Southern California, they made the rule that they wouldn't bring layers to Northern California anymore. That introduced some welfare issues in Southern California as the producers learned to depopulate on site and dispose on site. Northern California began vaccinating pullets before movement to lay farms, and they introduced movement controls for those birds. They introduced immunity monitoring. They learned a lot of things about what they were doing wrong. They ended dangerous practices on a voluntary basis around the industry. What's a dangerous practice? How about filling cages, during the life cycle of a lay flock with birds that, as they die over time, you fill those cages. That makes an open flock. So they closed flocks. They made the rule that you had to have closed flocks at all times. They held the turkeys on farms until seroconversion. So used controlled marketing to move those birds out. And the broilers. The broilers never broke before 35 days of age. Never, not once. They had to be moved to processing while they were shedding. So they had cross-commodity meetings held weekly to map their movements. Sometimes they went through school parking lots to avoid other farms. It was a massive effort, but it was strictly done by the industry. Who else knew? Who else knew how to map those movements when those movements happen? So, all right. So the outbreak then, once they introduced those controls and some others, the outbreak didn't end for another year. Okay. It was slow. You don't end an outbreak with immunization and a very large outbreak overnight. You're gonna have fewer cases and fewer cases and fewer cases, and then it ends. It's not one and done. The California industry learned that you had to monitor for immunity, determine infection status before moving, and movement controls are essential. They also learned that they had to work together, turkeys, broilers, and layers together with common goals in order to control the outbreak. So I asked our dear friend Bob Eckroad many years ago about this at the first one of the avian influenza symposia. And I said, how can we help the industry to end this? And this is what he said: when they decide they're done with it, they will end it. Why? They move all the birds. They do control the actual actions. So they have to be fully engaged. So vaccination must be a public and private partnership. The poultry industry, each individual commodity must be engaged to make a vaccination program successful. The goals of the program have to be fully agreed upon. And as an international group, we've heard some of the goals from different countries. We've heard we need to consider food security in some parts of the world. We need to consider trade. We need to consider the preservation of species. All of those things are at our doorsteps, and all of those things could be the goals that we need to discuss. Immunity may be difficult to achieve in the field. So Dr. Swain is going to tell you that maybe one dose of vaccine is enough. And Dr. Zavala is going to tell you, but I need 3 in the field because of immunosuppressive agents out there and stressors and all kinds of other things. And because the guys I hired to put those vaccines in went to lunch early and missed the other half of the flock. So to cover that, you have to achieve immunity. Vaccination and immunity might be the same in Dr. Swain's lab, but they grow very far apart in the field. Vaccination can be a tool of eradication, but only when everyone agrees. The expertise and manpower to vaccinate is controlled by the poultry industry. So only the poultry industry can immunize its birds, and only if they want to. engaging the industry for the private industry for collective good. Our poultry veterinarians with expertise are fully employed, doing all kinds of work. In order to vaccinate, that's additional work. And we're going to ask them to think about the collective good and not what they're hired to do, a singular company. So how can we do this? Do we build a task force? So we need to somehow engage those private resources. There may be lots of things that private industry and individual companies don't want to do, but we have to figure out how to advise them what's necessary and parse out what's not needed so that they can make those decisions and get engaged. In order to successfully eliminate virus, vaccination and immunity have to be equivalent in the field. The threat of high-path AI differs for each commodity, and thus triggers are going to differ. For the turkey commodity, which has suffered extensive outbreaks, the trigger has already been reached. I'm going to pause for a minute. I think you need to think about the fact this is our North American bird, the American turkey. This is, came from us. Our turkey industry has suffered greatly. Our turkey birds have suffered greatly. They're at the point of needing vaccination. We've heard from other countries that they are at the point of needing vaccination. And yet, if we pull that trigger for broiler companies who rely on trade, we've pulled the rug out from under them. The trigger that's used will impact the potential success of any mass vaccination program. So if we wait too long, we probably won't have any turkeys left. If we go too soon, we destroy the broilers for no reason. So how do we balance this? How can all the commodities be engaged to find common ground on vaccination? Well, we think that's possible, and we're going to talk about it in panel 2. So just a teaser. Regulatory agencies then, so if our private industry has the expertise, our regulatory agencies must be engaged and in the thought process of assuring that high-path AI doesn't circulate. We know that's going to cost a lot, so we got to get innovative. There's a lot of innovation out there, trust me. The private industry has a lot to do and will have a lot of controlling things, but providing a scientific basis for determining that the virus is not present in an immune population is essential for folks like Dr. Swain and other laboratories around the world. And that needs to be used by our regulatory agencies to control this. Maybe it's more testing, maybe it's sentinels. We've heard the downfalls of sentinels, but maybe there's another way. There's always a way. Controlling the outbreak has to be the goal. So consensus, in conclusion, consensus on the triggers and goals for the use of vaccination. We need conversations about what those triggers are going to be. When the turkey industry is in a corner, but the broiler industry is not, how do we get to that common ground about what those triggers might be? Implementation of programs using real experts, okay? Real experts. Those experts are employed in our country, in the United States, in private industry. We need to engage those folks to think about the common good. Field vets, vaccine manufacturers. You also are experts that, we need to engage for the common good. And we need to think about the fact that these individuals may have to undertake emergency vaccination or in a field setting under a global microscope. How do we protect them? How do we keep them from, I don't want it called the Cardona outbreak, okay, right? So how do we protect them? So surveillance for immunity and the virus in an ongoing and realistic way needs to take place. and needs to be part of the capacity that we build. The will and the funding to do it all. We cannot step off the curb and say, well, just put one toe in the water here. We need to put the whole foot in, and we need to all be engaged. So I heard the Dutch CVO talk about some next steps. I think we need to link hands and jump off that cliff together. In the face of a global challenge, we need to stick together. We need to remember what we already know. We know that mismatched vaccines won't work. How many more times do we need to prove that? We need to update vaccines. We don't. We don't need the field studies to do that again. We need to deliver them appropriately. And it's time, I'm going to say, from my personal perspective as an academic, looking in, I think it's time to rethink the undeclared war called trade and how we can come together to fight this scourge.
Thanks very much, Carol. Some great thoughts there on ways forward with vaccination and the issues which we're going to face. As we've seen, today's program is a little different to yesterday's, where our focus is predominantly on use of emergency vaccination. So rather than the preventive vaccination that we've been talking about yesterday, we're moving away from that towards what do you do if, for example, you are suddenly faced with an outbreak of this disease. And our next speaker, Ian Stageman, has been involved with this in a number of cases where he's used vaccination for other emergency diseases. And I passed it over to him for his comments. So Dr. Stageman is based in the Netherlands at Utrecht University, and I've known him for many years, working with avian influenza and other diseases across both Europe and Asia. Thank you.
Thank you, Les. So it's also a privilege for me to speak, to talk to you, to present. And I'm a veterinary epidemiologist, and well, having worked quite a lot on avian influenza, like Les indicated, I, as an epidemiologist, was, I think, considered by the organizing committee to be a bit less promiscuous regarding the diseases and the species I work with. So in that sense, I think I encountered quite a few of these diseases during my career. And well, this also gives me an opportunity to reflect on that. And well, actually the first one, and when I was a youngster, and I would like to start with that, was back at the end of the 80s when I graduated as a veterinarian. And after a short time in practice, I started working on a PhD project on Augeski's disease virus. For our American colleagues, this is called pseudorabies virus on the other side of the ocean. It's an herpes virus in pigs, and well, it was endemic to Europe and I think to many parts of the world at that time. And the general opinion in the 1980s, so at the time I started with it, was that vaccination was not a suitable tool to eradicate this virus. And the reason were actually quite the same reasons why we are so reluctant to start with vaccinating against avian influenza. That, well, when we experimentally challenged vaccinated birds, we could still infect them, and well, they would still excrete virus. And in addition to that, in the Netherlands and many other countries, there was a voluntary vaccination program where a great many farms were vaccinating against the disease. And this had not helped from preventing this disease to become endemic. In the Dutch situation, more than 90% of the pig farms got infected. And then I don't want to take all the credit with that, but at that time, my supervisor, Professor van Oorschot, and his colleagues, they had developed the first DIVA vaccines for this virus, which gave us at that time, I think, a great opportunity to start with an area-wide vaccination program where we really could monitor the effects of the vaccination in this population. And well, it gave me my PhD the thesis, which I made a copy of for you, so it's still available if you like, but it's already from 1995, so quite old. But it gave me the insight that vaccination does not need to be perfect to be an effective tool in infection control, because that's what it demonstrated, that it really could be effective. And it also gave me the life wisdom and that's also, I think, something for us here, that it's very often much more rewarding to think how something can be done than why it should not be done. And yeah, to finish this part on our Jeski's disease, I think the eradication of the virus in the Netherlands by area-wide vaccination took place. So in '93, we had over 90% ADV infected farms. By 2000, that had dropped to less than 0.1%. In 2002, the country was declared officially free, and in 2007, vaccination was stopped, and no outbreaks have been observed ever after. And I think this strategy has been implemented in quite a number of countries and has been successful. So it demonstrates that a leaky vaccine does not need to prohibit eradication. Good, and I think we have many other examples of this. This house where we are in now, and it's very nice that we can do this in the house of WOA. I think it's, for me, it's still OIE, and I'm glad that the old ornaments are still at the entrance for that. But well, it has been really instrumental in diseases like foot and mouth disease, which, well, vaccination has been very important on the worldwide scale. So picorna virus for cloven-hoofed animals, classical swine fever, not to be mistaken with the African swine fever, which is the threat at the moment, where also I think vaccination has been instrumental in the control of the disease. And then one that I think most of you, well, being at least the poultry people, will know is Newcastle disease by avian orthoavula-virus, which is also a name that is not, that I cannot easily pronounce because it has changed quite a lot, and so it will be changed back. But that's also one that you will know. So I'm an epidemiologist, so well, most of you that know me, you expect at least one equation in the presentation that I will give, and I will not disappoint you, although I will make it an easy one. Vaccination, from that perspective, is used in three ways, like we discussed yesterday. It's, well, it can stop transmission in an infected region, so that's how you would do it in an endemically infected region. Should you want to eliminate an infection, it can prevent infection in a free region at risk. That's maybe, well, a situation where a big part of the world is in now. And the other issue is then the emergency vaccination, where we stop spread in a previously free region upon reinfection. And for each of these, well, should vaccination alone be capable of doing that? Well, it should mean that the protection of an animal should be as high that each infected individual should infect less than one other individual, because in that case the infection will fade out. And before Corona, I needed to explain this always very extensively, but given the COVID epidemic, everybody has been drenched in it, so that makes my job much easier. But well, it's a tool that has been used a lot for this. And then well, to also make this comparison, because we heard that yesterday already from the presentation of Les, that for avian influenza, which is on the bottom of the slides, we have done quite a number of studies on that. And then we know that under experimental conditions, so the conditions that Dr. Cadona was referring to in David's lab, it's well, quite easy to have this situation where this reproduction ratio is below 1. So where vaccine by itself would be effective. But for conditions vaccinated in the field, that is not so easy. But for that situation, the avian influenza is not unique. Also for foot and mouth disease, we know that that is possible. And we know, for example, for certain that even in experimental settings, we cannot get this reproduction ratio below 1 in pigs, for example, which is one of the animals. Still, vaccination is considered an effective tool in the control of foot and mouth disease. Classical swine fever is maybe a bit different because I think the old vaccine for classical swine fever, the C strain vaccine, is probably the most effective, the single most effective vaccine on the world. Maybe people want to challenge that, but I think that's really a very good vaccine. But for our Jeski's disease, like I showed you, where we were capable of eradicating the infection, also there we know that in breeding stock, the reproduction ratio is below 1, but in finishing pigs, which is the majority of the population, it's still above 1, and still vaccine is capable of doing that. And also for Newcastle disease, we know that we can bring this reproduction ratio under experimental conditions below 1. But we know, at least for the classical vaccines, if we look at the HI titers, that really a big proportion of the poultry in the field does not meet the standards to obtain this. So there we also have the situation that vaccination as a standalone strategy will not be successful to eliminate the infection. But still, in those situations, it is a valuable strategy, and the reason is here. Because if the reproduction ratio is above 1 in the vaccinated population, introductions may still result in outbreaks, but the between farm spread can still be stopped, and because that's what's happening there. And the reason is that the proportion of introductions resulting in an outbreak will decrease in the vaccinated population, something also Les told yesterday. And that's the single equation that gives you an estimate on how big that is. So it's related to this reproduction ratio. Also the proportion of animals affected in an outbreak will decrease in that situation. The amount of virus excreted in vaccinated animals is decreased, as we can measure in the animals. And that means that, the between farm transmission is reduced. We should, however, keep an eye on that. And that means that in that situation, to be effective, vaccination needs to be accompanied by surveillance, movement control, and biosecurity. And that is what's happening with all these other diseases as well. Then to have a few other aspects to take into account while comparing these vaccination programs of foot and mouth disease, classical swine fever, Aujesky's disease, Newcastle disease, and avian influenza. The first one is related to, what happens if an animal has experienced the infection and has recovered. Well, for that situation, for example, in foot and mouth disease and classical swine fever, the situation is less favorable than, for example, for avian influenza, because if these animals can be infected, carrier status can exist. So that means that animals can seem healthy, but still be carrying the virus. And that's something that we don't know, neither for Newcastle disease nor for avian influenza, because if the animal is recovered, the virus, he has cleared the virus, so that is gone. So that's a good thing for at least the infection control. As for the major exposure source, I think that is also a bit different and maybe a bit more challenging. That for the terrestrial animal diseases, so the pig, cattle, and mentioned there, I think the major exposure sources are domestic. They all have a wildlife reservoir, and all wildlife is playing a role. But somehow we are able to take care of that. For foot and mouth disease, for example, I don't know sure about the situation in Africa, but for example, in northwestern Europe, the wildlife population is not able to maintain the infection, but that may be different in areas where you have a lot of buffalo. For classical swine fever, like already Tim Harder indicated yesterday, we have a suitable tool to take, at least to help the infection in wild boar by giving them oral bait vaccines. And for Aujesky's disease, they're also not playing a role. Whereas for Newcastle disease and avian influenza, I think we have also the problem in the wildlife that there is a third dimension in their movement. It's also easier for terrestrial animals because movement in 2 dimensions, it's much more complicated if you have this third dimension of a species that can fly. DIVA tests have been mentioned as one of the prerequisites for success of a program. Also there, actually, the experience with the other diseases is mixed. So for foot and mouth disease, it is being used quite a lot. For classical swine fever, it says no and yes. And the reason is that the most effective vaccine we have, so the C strain vaccine, is used quite a lot, but it does not have a DIVA in it. So that also means that it's not that if you have a really good vaccine, you could maybe do without. Aujesky's disease, I've mentioned that was really crucial to the success, but also for Newcastle disease, we have many vaccinated countries, but we do that without a DIVA strategy. For avian influenza, we have them. And then the final column here points to quick mass application of vaccination, which would be, I think, important for emergency vaccination. For the terrestrial animals, it's just injection, but of course, compared to poultry, the number of animals are relatively low, so that makes it often doable. For Newcastle disease, I think we have the big advantage that when we would want to do an emergency vaccination, we just go with our spraying and aerosol equipment, and we can do that pretty rapidly, getting sufficient titers. And that is, of course, a challenge for avian influenza, where at least in some parts we could vaccinate pretty quickly, but also a big part of the population, it would be a problem. I think initially the NDV vector vaccines were developed for that, but that may be challenging in these parts of the vaccine of the population where you have high NDV titers, because then the vaccine might not work. One word about trade agreements, because, well, some people get annoyed by the subtitle of this conference, which it says unnecessary trade barriers, whereas people say, well, why would you have unnecessary trade barriers? So we could rather talk about trade agreements, because I think at some point people, by their common sense, have made these agreements. And they are also for the other diseases. I think there are quite a few in place. So the reasons are that serological DIVA surveillance demonstrates the infection freedom in the past. So it does not tell you about the exact situation at this moment. And that is in particularly challenging if you have a continuous exposure from wildlife where you, actually don't know a farm that is free now can be infected tomorrow because of a wild bird virus incursion. So in that situation, for example, you could look in Europe to the NDV situation, where you also have unvaccinated countries and besides the majority of vaccinated countries. And there, I think many of the products can freely move. But if you would want to move adults or animals, you would have to put them through a quarantine. So that might also be a solution for avian influenza. As to the consumer products, I don't think that that risk assessments gave a lot of risk for that for avian influenza. In contrast to, for example, diseases like foot and mouth disease and classical swine fever, where these products have played really a role in initiating new outbreaks, just because the virus can maintain quite long in this products also related to the carrier status of the animals. So conclusions, to speed up a bit, vaccination does not need to be perfect to help eliminate the virus circulation. So we can dwell a lot about a perfect vaccine not being there for avian influenza, but trust me, also for the others, maybe the C strain for classical swine fever in exempt, also for the other diseases, there are no perfect vaccines. Then it still can be useful, as we've seen in the other diseases, if vaccination is accompanied by an adequate surveillance and biosecurity. But still, in trade agreements, these vaccinated animals will often have a different status compared to the unvaccinated animals. It does not need to be for the products. Then in the comparison, I think if we want to make progress, we could, I think, best look at the Newcastle disease vaccination programs, as there we have this contrasting situation. We have kind of a similar exposure, although of course different, but at least from the sources where we look on, we also do not have a carrier status like we have in the other terrestrial diseases, which makes it easier. And then we should address the differences that are there between avian influenza and Newcastle disease regarding, the opportunities for mass application, which really are different. But I'm sure that Ian in the next presentation is going to solve that for it. So I'm pretty happy that I'm before him in the lineup. For avian influenza, we have a DIVA test in contrast to Newcastle disease. So actually, we can much better see what is going on. And of course, there is the issue of the zoonotic potential, which is different for avian influenza than for Newcastle disease. But there, I think Richard Webby yesterday gave us a very reassuring, his idea that also from that perspective, vaccination would actually be better than not vaccination. So that brings me to the end of the giving, then a reflection. I think David yesterday gave us a very interesting perspective on the history of avian influenza. And I think that if we look at the current control strategy stamping out, the history of that is even longer. It goes back to the beginning of the 18th century and was first implemented for the control of rhino, initiated by the physician of Pope Clemens the Ninth, at least to protect his cattle in Italy. The Italian colleagues obviously know it much better. At that time, we did not know what a virus was. He did not know what a virus was. He did not have a test to find out whether the animals were infected or not. He did not have a vaccine to be able to protect them. Now, in these past 3 centuries, I think we are now at the point that we know every single nucleotide of the virus. We have tests that are highly sensitive, highly specific, and we have the results in a couple of hours. We have several vaccine platforms. We have ample experience with with the implementation of vaccine in vaccination programs with regards to other diseases, but we still apply this 300-year-old technique to control the disease. Whereas I'm sure that none of you came to Paris by carriage or by horse carriage or by boat. So we should be able to come up with a suitable vaccination strategy against avian influenza shortly, I'm sure, because otherwise I'm afraid that soon the current control strategies will be more devastating to the poultry industry than the disease itself. Thank you.
Thank you, Ralph. Wise words indeed. We're going to shift back to the issue of trade once again, and I'm very pleased to welcome Christian Wolf, who is the Councillor and Head of SPS section for the World Trade Organization. So let's hear what we have from the international organizations involved in trade. Thank you, Christian.
Thank you. Thank you very much. Trade has come up so much over the last couple of days, and I feel that I'm really on the spot here, and everybody is probably expecting me to pull the solution out of my hat. But I didn't even bring a hat, and I don't have a solution. So I will give you the perspective from the World Trade Organization, what basically just the activities, what is happening in relation to avian influenza. Solution is not with me. Probably some people here who know a lot about my area of work, the agreement on sanitary and phytosanitary measures, and others know very little, I imagine. So I just wanted to give a little bit of a background. When the World Trade Organization was established in 1995, basically the idea was that trade would be based on a set of rules that WTO members, which are governments, countries, agreed on. So it provides a legal framework, an international legal framework for trade. And the SPS agreement, the agreement on the application of sanitary and phytosanitary measures, basically is the international legal framework for trade in food and agricultural products. So it regulates actions by governments. It basically regulates the regulators to ensure that the impact on trade, for example, of regulations put in place to ensure food safety, animal health, and plant health, that the impact on trade is not more restrictive than is necessary for health protection purposes. So of course, yes, you restrict trade when it's necessary to protect, for example, animal health, but there should be no unnecessary barriers to trade is the idea. And so how do you distinguish between the necessary and the unnecessary barriers? Well, that's what the agreement is trying to do, basically reference to OIE standards or WOA standards now that are recognized as the relevant international standards for trade in animal products, and also the scientific basis of such regulations and so on. So I wanted to show you some statistics, some slides. One of the big principles of the WTO that is also incorporated in the SPS agreement is transparency. And what transparency means in this context is that when governments put in place regulation that will affect trade, for example, regulations related to avian influenza, they need to notify them to other WTO members, basically let their trading partners know that they're planning to do this when the regulation is still at a draft stage. And then we at the WTO, we make that available to all the other WTO members. And it starts a period where other trading partners can then submit comments on the draft regulation to avoid trade problems later on. So what I've put on this slide is, or rather what my intern has kindly put on the slide is a graph of all the notifications that we have, and we assign keywords. So we have a keyword for avian influenza. And the red bars are the emergency notifications, and the blue, you see below on some of them, some blue bars, those are the regular notifications. So that's supposed to be the normal scenario that, you know in advance that you're going to put in place a new animal health law or a new regulation for a particular risk. and you notify your trading partners, you give them 60 days for comments, and then at some point the regulation enters into effect. But when there are urgent health problems, members use emergency notifications. So here there's no comment period, no waiting before it enters into force. Usually they enter into force right away and are notified after. And that, of course, can be very disruptive for trade, but also very necessary. So we see that, and the green line, I don't know if you can read it, we've pulled from the WOA website the information on new HPAI outbreaks that are notified to WOA. And then you can see that it more or less tracks a similar pattern as the numbers of notifications. And it means, and you see the number of trade restrictions that have been notified, especially this year, has just gone up very steeply. So it is having a huge incidence on trade. And especially if you take into account that there are only 164 WTO members, and here you can see that there are close to 250 notifications. So you can see how many of such members are being notified. What is interesting to me is that most of them are emergency notifications, which means that every time a new outbreak is notified, it seems that many member governments have to take some sort of regulatory action and put in place an import prohibition from that particular country that has experienced an outbreak, rather than having a broader framework that allows them to take such action based on a legal framework that allows them to do so. Now, another piece of information I wanted to look at with you is what we call specific trade concerns. Now, at the WTO, we have a committee on sanitary and phytosanitary measures, and it meets 3 times a year. And all the WTO members are invited to come and to participate. And one of the things, actually, what they spend most of the time on at these SPS committee meetings are specific trade concerns. So the WTO as such, my colleagues and I, we do not police the measures that are notified. We do not check if they conform to WOA standards, if they conform to the SPS agreement, but members check each other. So they check each other's measures, and where there's an effect on trade, where there's a trade problem, then they can raise a concern, they can discuss it in the SPS committee. And you can see on the slide that we do have a steady number of such concerns related to avian influenza, mostly HPAI, in the SPS committee, but there we don't see the same steep increase. It's the same concerns that are reiterated over and over and over again. Yes, and about half of these well, we'll get to that. About half of these are eventually resolved, but others persist for years and years and years. And maybe just very briefly, some of the concerns that are raised in relation to these measures that are discussed are either that the restriction is not based on science, it does not recognize areas that are free of the disease, it is applied, for example, to the entire European Union when maybe outbreaks were only affecting certain countries or certain areas, or that the regulations put in place are not following the OIE standard, the WOA standard, sorry. Just here very briefly, again, the notifications, the large number of emergency notifications. And then this one is a little bit hard to see. But one of the things we ask members to include in their notification when they notify a new regulation that is being put in place is we ask them, well, is there a relevant WOA standard? And then is the measure in conformity? Does it basically do what the WOA standard says? And you can see that, especially for the emergency notifications, virtually all in 2022, 98% of them said, yes, our measure is in conformity with the WOA standard. But again, this is not policed by anybody except other members who may check whether they think this is the case. I we also looked in our database whether there are any notifications that have any reference to vaccines or vaccination, and that was only 12 notifications. So not very many notifications so far of regulations that deal in some way explicitly with vaccines or vaccination. So now just to give you an idea of the importance of avian influenza relative to other concerns. So this is, the pie is all the specific trade concerns raised in the SPS committee. And the blue part, the dark blue part is, well, all the blue parts are all the specific trade concerns that deal with animal diseases. And then avian influenza is 3% of all the specific trade concerns raised, and about, I think, around 10% of the animal health concerns. So these were the statistics that I wanted to show you. Of course, the number of specific trade concerns or the number of notifications doesn't necessarily say anything on the actual effect on trade. It just shows you something about the regulatory activity and the concerns being raised in the WTO. And maybe I should also mention that we have one dispute that has gone all the way through the WTO's dispute settlement system. That one was on an Indian measure on a requirement on avian influenza, was brought by the United States. And the SPS committee also has a mechanism to monitor the use of international standards. And there, one of the long-standing complaints every single time, every single meeting, is the non-use of the WOA standard for avian influenza by many members. So from the WTO perspective, the OIE, WOA plays a key role because its guidance becomes automatically the WTO reference point. On the SPS committee, there has so far not been a lot of discussion on vaccination. It's not necessarily, I think, the appropriate forum for it, but I think it also means that there's probably not a high level of awareness among trade officials, at least the ones working at the WTO. So I think there might be a chance there to raise awareness, either by organizing some sort of thematic session, some sort of information system session, just to alert trade officials to this problem and to give them the information. I wanted to also briefly mention that we have just embarked on a new work program called Responding to Modern Sanitary and Phytosanitary Challenges. And one of the big topics that is under discussion there is sustainability, sustainable food systems, sustainable agricultural production, and persistent obstacles to trade. So it seems to me that this would be a very pertinent case study to present in that context, and maybe the member states here present would want to think about whether it is something that they would like to raise in that context. Thank you, and that brings me to the end.
Thanks very much, Christian. Our final presentation for the morning session is by Dr. Ian Brown, who I don't think needs introductions to any of the members here. And Ian's going to be exploring some of the options for emergency vaccination. So what's on the horizon? What do we have at the moment, and what might be available for emergency use for control of avian influenza? Thanks, Ian.
Thanks so much, Les. Good morning, everyone. So I'm going to go through quite rapidly here, keeping you from your coffee break. So I think it's worth just reflecting first. Emergency vaccination. Well, it's an option where there's evidence for AI introduction, and that can be in many guises, as Les indicated yesterday. But generally, you'll have epidemiologic evidence that will support perhaps massive introductions. So we know the wild bird risk is creating that constant level of threat. So you've got two options. It's protective, so that's vaccination to live, or suppressive, which is vaccination to kill, which obviously is probably used in more extreme situations where you have something that's fast spreading. But of course, if we look at the protective option, we know that that requires monitoring and surveillance to detect infection in vaccinated flocks. So we need the right tools. Of course, you want an efficacious vaccine. you want to be able to deliver it on scale to large numbers of birds. Preferably, you do not want it to transmit from flock to flock, but some replication within a flock to enable broader flock immunity is an advantage. And of course, we can achieve this using a variety of vectors. Of course, it needs to be safe. We've reflected already several times. We do not want anything to revert to virulence, and we've got the problem of genetic reassortment. So at a farm level, you could envisage a situation where you could have a break in one house and you could vaccinate the other ones on an emergency basis with spray or live vaccine, or of course at population level, you may target that based on risk and the sector type. So I think we're all familiar with this. There are several ways we can do this. Traditionally, of course, spray vaccination has been used for a number of poultry diseases, as has drinking water. Feed is attracting some attention. And of course, we have the possibility to do mass vaccination in ovate hatchery. It's worth remembering, as Ian's already reflected on, that we faced these questions many decades ago. Well, not myself, but my predecessors in terms of Newcastle disease. And whilst it's different, you know, there were questions there about how they would do it, how they would overcome MDA, et cetera, et cetera. So, you know, this path has been charted to some extent before. If we look at the utility and type of vaccines, I think the most striking thing is there are lots of options. We are not limited. In fact, perhaps we've got too many options. And, you know, from whole inactivated virus, which of course is a major use vaccine now for AI. We've got live attenuated virus-like particles, recombinant bacterial vectors, DNA, recombinant viral vectors, and subunit proteins. So there's an awful lot out there that we can do. But we do recognize that inactivated vaccines for mass application have very limited utility because they don't induce sufficient protective immunity, generally speaking. Of course, there are live attenuated vaccines for many avian virus diseases, but of course, they haven't got this problem of risk of reassortment. So any vaccine, I think we'd have to accept, needs absolute surety. So there's a number of principal options, but of course, really the ideal is to be able to deliver these vaccines to the mucosal surface. That's where the virus first enters the host. That's where it's going to attach and replicate. That's where you want your protection, ideally, to be delivered. So I'm going to briefly look at some of these options: live virus, live attenuated, inactivated vaccines for aerosol delivery, viral vector vaccines. We've already heard a lot about those, recombinant proteins, bacterial vectors, plant-based vectors. And then there's some things that we can do now in terms of upregulating the immune responses, all sorts of creative opportunities. So I think in looking at this, it's actually quite staggering how much R&D is out there. I work in the field, and I was genuinely surprised about how much is out there that we can learn from and apply to AI. So some of these things are a bit blue sky. They're a long way from market, but I think some of these things actually have got the opportunity to take forward. So I think the purpose of what I'm going to show you now is not so much the detail, but just an awareness of actually what the options and what things we can do out there. So I think let's put the influenza live wild type to bed. I will come back to it briefly, but I think we accept that's not necessarily going to be what we want to do moving forward. And do we actually need to? Of course, a live vaccine's got the benefit that it's a much stronger potentiator of the immune response. And there's some very nice technologies out there for modifying live viruses. And of course, they've got the benefit that they look pretty much the same as the live virus itself. And we can do things now to manipulate the genome to make those viruses safe, less virulent. We can attenuate them. We can modify some of the proteins that direct infectivity. We can do things like adapting those strains to operate at a temperature that's not the normal body temperature of the bird. And we can manipulate the hemagglutinin. I mean, this is well established that you can modify the hemagglutinin. And then, of course, there's the vector vaccines, which we know have lots of advantages. And of course, the challenge there is how many proteins can we stick in our vector that give you a broader protective response? So a low pathogenicity H5 virus. I know this was looked at when Goose Guangdong first emerged. We had a virus in our lab, H5N3, Duck Singapore. And there was a lot of interest at the time because we were dealing with a completely new disease. So there is data out there that says you can vaccinate with a low path virus in drinking water, and you can get protection. Of course, the big problem with that is licensure for a vaccine such as that, because clearly there'll be a strong possibility of mutation to virulence and reassortment. So if we look at the live attenuated vaccines, I've already mentioned that there are techniques now. So the cold adapted technique is a technique that human influenza virologists have been using for many years. They take a virus and they effectively adapt it to work in conditions of low temperature. So this attenuates the virus, but it still, of course, behaves like a normal virus in terms of being able to induce a very broad range of immune responses. And of course, we've got H7s as well. And we know some parts of the world have been vaccinated against H7N3. And there are studies that have shown, again, a Dutch study showed that if you take an H7N3 virus, you mix it with one of these donor viruses to make it cold adapted, it can replicate well in the chicken. But again, you come back to the problem of reassortment risk. So we know that you can modify the cleavage site and you can modify it to a low path virus. And in fact, if we take the non-structural protein, we can modify that as well to attenuate the virus. And by manipulation, you effectively reduce its replication efficiency. And of course, these viruses in experimental studies are shown to be really effective in offering a broad protective response to lethal challenge with H5N1. Now, this is an interesting technique where what the authors have done of this study is they've taken the H5 hemagglutinin gene and they've inserted it into a part of the genome where it doesn't normally reside. So they put it into an H9N2 virus. And the reason why this they can consider to be safe is because it reduces this possibility of genetic reassortment, because the gene is effectively buried in another gene segment, but it's still fully expressed. So you still get a broad protective response. So this is an interesting approach. I'm not sure anyone's following these approaches up, but certainly interesting data. Now, of course, one of the things that's actually made people even more nervous about live attenuated vaccines is an experience our American colleagues had where they were vaccinated against swine influenza. So, okay, that's not a reportable disease, but nevertheless, it was a major problem. And they used the live attenuated swine influenza virus. It was introduced about 2017 into pigs. And about a year later, through surveillance, it was noted that there were reassortment events occurring with endemic swine viruses. So they were mixing and switching their gene segments. And that map there shows you a lot of genetic diversity. That, of course, caused a lot of concern because it was adding to and making the problem worse potentially. Of course, if you use such a product in an emergency basis, then obviously, and it was a short, sharp application, then maybe that opportunity to reassort is much reduced. However, it still fundamentally is probably the best evidence we've got at the minute around the risk and that we need to be aware of it. So there has been a bit of work done on inactivated vaccines for aerosol delivery, but generally speaking, as I said, these don't potentiate such a good response. But there's a nice study here where they actually use a spray freeze-dried product, and they were able to show that pulmonary vaccination by inhalation did actually protect, and they were able to further enhance that by ensuring that birds in contact could inhale the dry powder, and so therefore you've got a potentiating effect. And then they enhance that by using bacterium-like particles. So again, does this have possibilities? I think that this was an immunogenicity study. Would it really protect against a lethal challenge would obviously need to be shown. Now, another approach, which is probably a logical one, is to use vectors based on avian paramyxovirus, but instead of Newcastle disease, and we've heard a lot about MDA and the problems there, is there's some new studies now that have come out that have used avian paramyxovirus serotype 2. So we know that that's found from time to time in poultry. It's got a replication competence. So here you use that to deliver, in this particular study, H9N2. So they took the hemagglutinin of the H9 virus, they put it into the avian paramyxovirus serotype 2, and they were able to protect chickens from challenge with H9N2. And there's a more recent study that's applied the same approach with avian paramyxovirus type 3. So they took the hemagglutinin of H5 and they inserted it into the PMV3 genome, And again, they got very good neutralizing antibodies and protective effect. Now, there's another interesting study that's come out of our colleagues in Germany at FLI. And what they've taken is a bat orthomyxovirus. So these bat viruses are quite different. They don't behave biologically the same as influenza viruses in birds. And they've got this subtype H17N10. It seems that when we look in bats, we might find more diversity in there. And this virus doesn't want to be in a bird. So what they've done here is effectively they've taken the hemagglutinin and the neuraminidase, because we know there's a benefit for using both of those glycoproteins to get good, strong protective immunity. And they've effectively inserted H5 and N1 and made this chimeric virus and then done a challenge with H5N1. And you can see on the right there that they got complete clinical protection. So this is a very interesting approach, because this genome would be not able to reassort between a normal avian influenza. That's the theory. I mean, there's obviously more work needed in this space, but it's an interesting approach. And then, of course, there's a range of other vectors that we haven't got time to go into, and Erica Spackman touched on some yesterday, with the adenovirus. But of course, the big problem with all of these is that you might have MDA, and you need to know your target population and what that background immunity might be. Then there's drinking water. Well, of course, we know this has been well used for Newcastle disease, but there's some interesting work where you can take a salmonella vector, in this case, Salmonella gallinarum, and you can insert one of the core proteins, the matrix protein, and simultaneously protect against fowl typhoid and H9N2 in this study. So can we use bacteria as a delivery vehicle, which will then aid mass vaccination through drinking water? Similarly, lactobacillus, there's some studies here where, again, they've inserted the matrix protein and the hemagglutinin. So notice you get a double effect. If you can insert more than one protective gene, you can broaden the immune response, which gives you a stronger protective effect on challenge. Of course, there's other benefits that if you use oral vaccines using bacteria, you can upregulate some of the immune responses. So co-administration or oral administration, so in this case, typhimurium, you can upregulate chicken interferon alpha, which of course broadens your protective response. So there are other ways that you can modulate this design to not just protect against the key proteins, but actually to upregulate parts of the bird's immune system. Yeast, I think wherever you look, there's all sorts of platforms that have been used here. This particular study used yeast, and it was the H7N9 virus that we heard about from Les yesterday, where obviously they vaccinated to control. This is about expressing the hemagglutinin. Of course, the advantage of these systems is they are relatively low cost once they've developed in terms of the technology to produce very high quantities and potent doses. So plants are getting quite a lot of interest, not just in this field, but for other diseases. So a plant expression system enables you to maybe produce subunit vaccines at very low cost, with relatively simple infrastructure. So effectively what you do is you insert your target gene into these plant expression systems, and they produce that protein. The key thing is, whether you get the conformation in the protein, is it presented to the bird's immune system like the natural virus? Well, there's some studies here that are showing using cowpea mosaic virus, they've inserted the hemagglutinin, and they've been able to show that the conformation shape of that hemagglutinin is faithfully reproduced, and therefore induces clear immune responses, maybe not protective. The problem with these systems is they need tweaking because they're quite low in immunogenicity. Likewise, there's a further study here where they use the matrix protein, that's the core protein, and they put that into duckweed. So if we can use these sorts of delivery systems, then we've got the possibility to potentially deliver vaccines through feed. I've talked about agents that stimulate innate immunity, and of course that's really important when you're talking about mucosal surfaces. And we have known for some time, some parts of the world have observed, and indeed there's experimental studies, that because there is a similarity in the H9N2 genome compared to the H5N1 viruses we're currently plagued with, we can use that to prime parts of the immune system that provide a protective effect. So it's been noticed that in naturally infected H9N2 flocks, you can have an attenuation when there's exposure to H5N1. And we can use these approaches to upregulate parts of the immune system, such as toll-like receptors, which basically enhances the ability of the immune response. So there's all sorts of gizmos and technologies that are well beyond my comprehension now, using nanoparticle platforms in how we can present these antigens to the bird's immune system to get more bang for our buck, I suppose, is the message. And of course, we need to look at other systems. So there's one here from dogs where they've looked at how they can upregulate these toll-like receptors in immunotherapy in that host. And then, of course, vectors have other benefits, that they can have switches, if you like, to regulate the immune system, so interferons. So this particular study here is looking at foot and mouth disease, and they're able to induce strong antiviral responses and adjuvant effects. So you can design your construct not only to provide protective immunity to your target immunogen in your virus, but you can actually upregulate parts of the bird's immune system. Again, this is a toll-like receptor. This is a novel mucosal inactivated vaccine. So note, this is an inactivated vaccine. There is opportunities here to use these receptors to activate dendritic cells, which broaden and potentiate the immune response. Again, use of bacteria. You can insert influenza proteins into the bacteria, which have not just the benefit of providing a protective immune response to the virus, but they can upregulate elements of the immune system. And this particular study here claims a single shot can produce, well, it can induce good immunity. Whether it's protective is something this study didn't go as far as examining. And there's another approach here with Salmonella typhimurium, where they insert the hemagglutinin globular head into the flagella, and that can potentiate and broaden the response. So sort of finally, we're now in an age where COVID has taught us a lot. And I think there's a huge body of learning from COVID, and it's changed everyone's mindset about how we vaccinate. So we've already heard at this meeting about mRNA vaccines. And there's a recent study here that obviously was looking at these in terms of the S protein, but equally we could substitute hemagglutinin. And this, of course, has the benefit that the host cell translates that protein and presents it to the immune system. The induction of strong responses is something that needs to be adapted and improved probably, but basically you get a T and a B cell response, and there's very good protective immunity. So I guess the question here is, can we take this new generation of intranasally administered technology to HPAI and get that good protective mucosal immunity? And I know there is work that's looking in this space. This is the next generation of vaccines. And if we're talking about, I don't know, maybe 2-3 years, maybe these vaccines are going to come to market. They have the advantage that because they work off what we call a negative strand, we have less risk here of the genetic reassortment problem. And then just finally, recombinant protein technology, not to ignore this. This is the technology that's been around for a long time. Purify the immunogen, and we can do that in all sorts of ways. You can express it in human cells, like in this particular study here. You can do it against one protein or against 2 proteins, and you can express these proteins in E. coli and make very large quantities. So there is use of this technology. Can we adapt it and use it on scale for influenza? So I think to wrap up, that's just giving you a snapshot that there is substantial research effort out there. So in the academic world, there's an awful lot of studies being done, and I think they're possibly going under the radar. And we heard yesterday, from our colleagues in pharma, and it's how we can bridge the gap between that academic research possibility. How can we take some of that into the marketplace for next generation vaccines? There's an awful lot of options out, and of course it's focusing on the right pathway that provides the right opportunities. But I think there is a wealth of information out there, but most of these studies are 1 or 2. A research group produces a paper, they maybe go and refine it and publish a second paper, and that's where the work stops. So I think we've got to challenge ourselves and think about how can we take some of those promising options into the marketplace, which would of course attract a regulatory approval and would be safe and efficacious. So I think, you know, to finalize, it's like Ian said, we didn't come here in a carriage. We've now got a toolbox at our disposal that we haven't fully unlocked. We've done it for COVID. We now have the possibility to look at that toolbox and go through those lessons and see how they can be applied to HPAI. So thanks for your attention.
Thanks, Ian. Terrific food for thought. We've still got a few minutes left before we break for coffee. So if we have any questions from the floor, does anyone have a question they'd like to ask any of the panelists? We've got one here from Carmen. Sure. One from Carmen that's come via the online chat. So
yeah, this question is not from me. This comes from Nick Eteradossi. And he is asking to Ian, the lack of career for AI and ND, do you mean only in recovered animals, as some inapparent infections could indeed occur, especially in dogs? So this is really a nasty question.
Not sure I fully understood the question.
You talked about the lack of career for avian influenza in Newcastle. And Nick, he is asking if you mean the recovered animals only, or some inapparent infections which could indeed occur in dogs.
Yeah, so what I meant there was the well, it's kind of a definition question. So for avian influenza, for Newcastle disease, birds experience an infection, and then at the end they recover because they have immunity, and then, well, if they have sufficient immunity, then the shedding stops and the bird is recovered. And the person of the question is completely right that there is difference in the length of that period between species. So for chickens that will be shorter than for duck, because for ducks it can be quite long. But if you go to, for example, foot and mouth disease, after the infection, and there the carrier status is defined 28 days after infection, then well, in the unvaccinated situation, but also for a lot in the vaccinated situation, in half of the animals still the virus can be found in probe bank samples in the throat of the animals, which is then there defined as a carrier animal. But in classical swine fever, you have even a more complicated situation, that you have, on the one hand, a chronic infection status in which the animals do not really recover, but become chronically infected, which can be for a very long time. And there's also the option that intrauterine piglets can get infected. They don't get an immune response at all, and they become what we call immune tolerant carriers. So they become seemingly healthy to the world, and but they are still shedding the virus, and they can do that for a very long time without being seen. And these phenomena, which are apparently for the survival of the virus in the other species, we don't see them in Newcastle disease and avian influenza, I meant. And for the herpes viruses, as you probably know, like our Jesse's disease, it's the latency that you have in your neural tissues, the ganglia, that makes the virus a carrier. So it's a definition issue, and well, this was what I meant.
Okay.
we have one more online question.
Yeah,
You'll have to pause.
And in the meantime, another question popped in. This is for Christiane Wolf. And I tried to read it, but it's very long. I hope if there is an understanding. VOA already allows for vaccination, yet you have heard the main resistance to this trade. You have also heard that there are very few reasons a country should not be able to come up with an effective, implementable and safe vaccination program. So what can this group meeting here do for WTO specifically to provide the assurances that a vaccination strategy does not result in severe trade issues? Because it would be nice to have a broad and strong WTO statement on this topic, to hopefully get in front of any potential trade issues, or at least give a strong backbone to member countries that would decide to embark on allowing this valuable tool to actually be a usable tool in our toolbox. That means really do not punish the people who try to the countries who want to jump in the pool, as we decided yesterday.
Yes, thanks. That is a long question. Well, a number of observations. The first is that the WTO as such is completely agnostic on any scientific issue. We are lawyers and economists. We do not try to set the standards that are applied in international trade. So the way it works is that, as I mentioned before, the agreements provide a legal framework. And in the case of the SPS agreement, it contains a reference to international standards. So it strongly recommends that members base their animal health regulations on WOA standards, but members can deviate from those standards. So for example, if a country wishes to achieve a higher level of protection, or maybe no relevant standard exists for a particular situation, they can, of course, put in place a regulation based on a risk assessment in that case, on their own risk assessment. And there are certain requirements for what a risk assessment must do. It must take into account the relevant science and so on. So there is a strong incentive to use WOA standards. If a measure is basically the same as a WOA standard, it is presumed to be in conformity with WTO agreements. So there's a certain level of protection from legal challenge. But still, you see that many members deviate from international standards. They do not necessarily apply them. And that's why we have a lot of discussions in the committee about members asking each other, why are you not using the WOA standard? So I think basically, unfortunately, you don't have to convince the WTO, but I think you have to convince each other. You have to convince the veterinary authorities of other trading partners to accept vaccination. As I think you said, you have to jump off the cliff together, probably, and convince each other to accept it. And I think what you could do at the WTO, as I said, is try to sensitize or raise awareness among trade officials, the ones that come to the WTO. But I think probably the more important work is to be done among veterinary services in convincing each other. Thank you.
Thanks, Christian. Thanks for a great session this morning. And I'd like to again just have one quick question. I know we use our breakout sessions to do that, so we're running out of time. But one very quick question, Peter.
It's a question for Ian. When talking about emergency vaccinations, and when you compare that, for instance, with Newcastle disease, we know that we vaccinated all our birds already, but then still, when there's a danger and there's Newcastle around, we still go in with an anatomist and vaccinate them again, mass application. And although there is a lot of immunity around, we still see that there is a raise in immunity and that you get extra local immunity, and that really helps in preventing the disease. When we're talking about this recombinant with Newcastle, then the question would be, okay, if there is somewhere an outbreak and you would like to do something in the emergency situation, then still you could use this kind of vaccination technique with anatomist and get the local immunity working, and then also against the influenza. Would that be something that you could imagine?
Well, if it's possible for Newcastle disease and it's safe to do so, I wouldn't see any reason why you couldn't. I mean, we know that, you know, as you've alluded to, some of these responses have a pretty short half-life, so they can wane quite quickly. So hence the fact that that potentiates it. I mean, it's getting that level of protective immunity across the flock, isn't it? And not having birds with low immune responses. So I don't see a block as to why, if you've got a safe and efficacious delivery system, you wouldn't be able to use that approach for AI. But I don't know. Do others want to comment?
Yeah, so probably because it's a good point, Peter, and I think it surely would work in those situations where you have, well, kind of a poor protection. So in broilers, I'm sure, because we know that we have very low HI titers against ND. And so maybe by combining it by, for example, some kind of pre-testing on what titers for ND are present in the farm and vaccination, you could also maybe a priori already estimate how effective it would be, but it would certainly be something at least to keep in the toolbox and to implement in those situations where it could be useful.
Thanks, Anne. Thanks, Ian. Thanks, Peter. Okay, great session this morning. Thank you all. Coffee time now. I'd just like to thank our speakers one more time.
So we're on to the next panel session. And you've all seen how the panel sessions work now. So hopefully nothing will be too much of a surprise. The topic for this panel is emergency vaccination in a mega layer farm in the face of an outbreak in a country that is or was free from infection. So again, this is the emergency vaccination situation that the panel has been assigned. So what were the objectives given to the panel? First, to develop an emergency vaccination program for large farms in the face of an outbreak in a country that was or is or was free from infection and wants to regain this poultry freedom. They want to consider the options for preventative vaccination of high-risk flocks and emergency vaccination on affected or at-risk farms. And they've been asked to identify solutions that would allow a return to infection freedom in a time frame similar to a stamping out process. So those were the objectives that were assigned to the panel. Of course, when you go through panel discussions, these things evolve. We'll find out where they came to. And I hear Carol laughing in the background. Now, this panel's been working really hard. You saw them right before the start of this presentation, just finalizing the presentation. And I'd like to introduce them very briefly. So first of all, we have Dr. Julie Gauthier from the USDA, Department of Agriculture of the US. We have Dr. Matthias Voss, an EU poultry veterinarian and a member of the study group from Germany. We have Dr. Carol Cardona, who you've already met, has been introduced earlier. We have Dr. Geeta Srinivas, who's from the USDA Center for Veterinary Biologics. This is a new approach that the panel is taking. They wanted to be a little creative. So actually, Geeta is just up in the mezzanine, but she's pretending to be here through electronics. No, actually, she's back in the United States. And then we have Dr. Peter Wingen, who is a private poultry veterinarian out of the Netherlands. And finally, we have Dr. Guillermo Zavala, who is a private poultry veterinarian who covers Latin America. So the panel process will be the same as yesterday. Okay, first of all, we're going to have about 50 minutes of presentation, dialogue, and discussion. And then the panel will ask for any clarifying questions to prepare you for the breakout sessions. Once those 50 minutes or 55 minutes are up, then we'll do the breakouts. The breakout will be essentially organized the same as yesterday. And we'll put that information so everyone can remember where they have to go. Then after the breakout groups, where everyone will discuss the topic and work towards identifying solutions to the issues or barriers, we'll get back together. We don't have a break in between this time. We'll get back together and the presentations from each of the representatives from the different breakout groups will occur. And again, I'll remind you, elect, appoint, or coerce a spokesperson to start capturing the information so they can share it with the group. Each person will have about 10 minutes to give those presentations, and then the panel will go through and we'll ask discussions and topics of concern from the panel's perspective, and then we'll hopefully have time to do questions as well at that. Okay, so we'll start in 1 second. I'm going to actually stay up front. I'll try to stay out of the way but because of the way that we're doing this, Dr. Srinivas has visibility, but it might be difficult for her to communicate back without creating an electronic loop or something. So she will tell me her comment, and then I will share her comment. So I'm sorry, but I will wander around a bit up here. Carol, it's all yours.
Thank you. So just to start you out, I have to tell you the slide that's up here. We'll get to this in a minute, but you know, this slide explodes and continues on forever. And this is basically a summary of our notes from our meetings. And the reason that it happened this way is because we each found that we brought a very different perspective to the issue of vaccination. So I'm going to introduce myself first. So I'm from the University of Minnesota, but I'm going to tell you that I have a very practical kind of ground up approach to looking at vaccination. And so that's sort of the perspective that I brought. And so many of the issues I thought about were way on this end of the slide. Julie.
I'm a veterinary epidemiologist, or I pretend to be one, and I lead the team with APHIS, USDA APHIS, to develop strategy and policy for poultry health programs. So since February 8th of this year, we've been actually implementing scenarios like we'll present to you today, and having to come up with solutions over a weekend or overnight and putting them into place, regardless of some of our concerns and barriers. So we'll point that out, and I really welcome your discussion, and would appreciate you poking holes in the ideas that we'll present to you, because this is how we can form a plan and actually get to the point where we're using vaccine.
Yes, I'm Matthias Voss. I'm veterinarian working for primary layer breeder company in Europe, in Germany. I have been also president of the Poultry Veterinary Study Group of the EU. And as it was mentioned yesterday, we made a position papers 2 years ago about vaccination, to start the discussion about vaccination and to find some solutions there. So we may ask some strange questions, or because the scenario is related to the United States. So we have some different conditions in Europe.
And I'll say that I think Matthias and Peter are both playing the role of skeptic on this particular panel.
So yes, I don't want to be rude, Carol, but I'm going to ask you some questions. So I'm Peter, not Wingen, but Wijnen. That's the plural of wine. So,
Sorry.
So, but I'm a private veterinarian and working only with poultry. I've also been president of the PVSG. It's the Poultry Veterinary Study Group of the EU. So all countries are involved there with all poultry veterinarians. So we have got some nice discussions going on, also about AI. I'm very pleased to be here, and I really hope this brings us forward towards vaccination.
Thank you. I'm also pleased to be here. My name is Guillermo Zavala. In my career, I worked for 2 vaccine companies, 1 breeding company, 2 diagnostic labs. And my last formal job was with the University of Georgia, where I was a professor doing research in virology, specifically on retroviruses and laryngotracheitis, and where I also did clinical extension service for the state of Georgia industry. And of course, I also did teaching. And today I do private consulting in many countries, not just Latin America. I've done work in some capacity in some 65 countries.
Great, thank you. And Geetha. So just give a brief bio on yourself, please.
And perspective.
HPAI and what can be done moving forward, what technology is out there, and how quickly we can license these vaccines. So such things. So that's my background.
Perfect. So I'll do an abbreviated introduction. I don't know if everyone could hear that, but this is Dr. Geetha Srinivas. She is a veterinary. She works for the USDA, the Center for Veterinary Biologics, which is the organization that regulates vaccines, veterinary vaccines in the US. In her role, she's active in supporting the registration for the development of vaccines, such as vaccines for avian influenza.
Thank you, Geeta. So we are, as we did our discussions, I think you can see that there's a lot of work to be done to get to the actual delivery of vaccines into birds in an effective way. There's a lot of questions that we all have, but at the base of that question is the decision to vaccinate. And we felt like that decision to vaccinate, unless it's taken, that the rest, it sort of acts as a dam and we can't move forward, or we won't move forward. So to quote James Carville from the Clinton campaign, it's the trade, stupid, right? So we aren't going to make that decision to vaccinate unless we get over that trade barrier. To get over that trade barrier, we have to talk about all of those things that are dammed up behind this decision to vaccinate. And we have to come up with a proposal. So we eliminated the preemptive vaccination scenario for you. We've already opened up trade because otherwise we can't get to the place where we need to talk about what Julie talked about, that on the ground actual scenario that we would face and that we want to make you face. All right. So our post-infection or our emergency scenario, we have a growing outbreak. There are now more than 15 infected premises in your area. In our case, it would be a state. The virus is spreading from farm to farm, and wild bird introductions are continuing. Stamping out has worked so far and is keeping up, but resources are becoming limited. People are tired. We have no available crews right now, none. So the next densely populated area is of great concern. We think that at this point, it will be too much. We can't handle it. So vaccination is an option for premises in the 10-kilometer control area. There are 66 farms in that area. There are 12 commercial broilers, 51 commercial turkeys, grow out barns. There's one large lay farm with 5 million hens and 2 pullet farms with 500,000 birds each. Okay. No, they're in control. Okay, so here's your picture. Thank you, Julie, for picking Minnesota. I'll let Julie explain.
Oh, I just wanted to give you a picture of our 10-kilometer area it is Minnesota, but we'll protect the identity further. The purple flags are with O's, those are open investigations. So this was just what's currently in our emergency management database. And the C, the lone C, is one closed investigation. So you can see that this thing is still ongoing.
Okay. So all of our people are out in the field, or all of Julie's people are out in the field. Everybody's concerned. All right, so our emergency vaccination proposal for the 58 stocked farm. So there are 66 total, So 8 of them don't have birds, right? Okay, in the 10-kilometer control area, we propose as a panel, right? We're all together, actually, skeptics together. We would not vaccinate the birds currently on the 10 broiler, 45 turkey, and 1 lay farm. We think that we don't have the ability to vaccinate those populations. That the risk of bringing in the people to do injections on those farms is too great with the level of active infection that's going on. We propose to vaccinate the pullets before they would move to the lay farm. Why would we choose the pullets? The pullets are, we have the ability to vaccinate pullets in place on those farms. We have people who normally vaccinate. The pullets would be immunized once, right away, everybody. They would be vaccinated at another time, twice or thrice, depending on their ages at 8 weeks to 10 weeks-ish, 12 to 14 weeks, depending on when those birds are going to be handled. So we coordinate it with what already happens with them. Turkeys being placed in the 10-kilometer zone would also be vaccinated. So if they are going to come into the 10-kilometer zone, when turkeys in the zone are marketed out, then we would replace them with vaccinated birds. They would be vaccinated in the brood, in the hatchery and in the brood stage, but not in grow out. So pullet vaccination. The goal of the pullet vaccination is aimed at protection of the very large lay farm that they supply, okay? So 5 million birds, we open up the farm, we open up the flock when we place pullets. We break the line of separation, we bring in people and equipment. At that time, our lay industry in the United States has a big problem. We have a hard time mapping where the people who do those moves have been and controlling that many people. So we propose to protect those birds from the challenge they might get during that movement. This is a critical and unavoidable breach in biosecurity for these farms. Vaccination would improve resistance of the pullet flock if they're exposed during the move. The newly placed pullet flocks will be segregated on the lay farm for 2 weeks post-placement following, normally talk about in the, they talk about in the layer industry, a salmonella protocol. Okay, oops. The 2 pullet farms in the 10-kilometer area and 1 additional farm that is 40 miles or 64 kilometers away supply pullet to the lay farm that's in the zone. Pullets on all the farms will be vaccinated as soon as the detection is made and the decision to vaccinate is done. The vaccination will be at the hatch first, everything gets vaccinated. Then we go into the normal vaccination protocol, which would be at hatchery, either at 8 weeks or 12 weeks or both. It would be coordinated with existing vaccination schedule when birds are already being handled. That would be the producer or the producer's veterinarian's choice. Vaccination will end when the control area is released plus 28 days. So that's about 63 days or 9 weeks. Okay. Pullets will be vaccinated a minimum of 2 weeks before they're moved to the lay house. So what if my birds are 18 weeks and I have to move them today? The industry would have to cull those birds, or they have to have good light men who can keep them from going into production. Okay. As I said, vaccinate everything on the farm as soon as possible and continue with scheduled vaccination as the birds age. So what would be possible for those birds that haven't been placed? You'd have potential for 3 vaccinations for the 4-week-old birds. You'd have 2 vaccinations for the, et cetera, et cetera. Okay. In the scenario that we're talking about, because the control area will be released before those vaccination schedules will be fulfilled, you'd likely only get 1 round of vaccination into those pullets. Okay. Hello, my skeptical friends. So if we think about this scenario, talking about a 50 house layer farm. Okay. Each house has 100,000 chickens in it. They're connected with an egg belt that goes directly to the egg processing plant. There's an on-site, and then eggs or egg product flows from there. There's a feed mill on site into which feed ingredients flow and is delivered by a conveyor to all of the houses. Okay. So when we vaccinate our first pullet farm, we would place 1 flock on that farm that's vaccinated, and that's where that flock might be. So, panel, do we all agree this is a good idea?
No, I was so amazed about this. Talking about a farm, 6 or 5 million birds. That's 1/6 of the amount of animals anyway that we have in complete of the Netherlands, whole of the Netherlands. So it's incredible this. And then you have such a farm in an area where you have an outbreak. I think we would go for a risk analysis here. And this will take about a year before you vaccinate the complete farm. When the infected farm is in that zone, Jesus, I'm worried, especially also about all the trucks going in and out. So I think I asked myself whether this is feasible. So that was my critic, but okay, we're going on. Maybe you have some comments.
How would you manage this facility then? If
I think we wouldn't. I think after the risk analysis, they would say you have to kill them.
that comes with a $16 million price tag, having been through
And
that.
this farm has been surrounded before by diseases that it never got. So,
Well, I have a comment. This bug, avian influenza, is so infectious, they will get it. I think the risk is too high.
Okay, you just demonstrated that this layer farm is relatively good protected by biosecurity. So
Yes.
the biggest risk is to bring in the pullets,
yes.
which are vaccinated with something we don't know in the moment. So we bring in birds with the highest risk of being infected due to handling of the people. And we put a huge challenge on these vaccines also, and the reputation on what we are discussing here. We want to implement vaccination in the control, and we have a scenario where there is very little chance that these vaccines can fulfill what we expect from them.
Are you saying that if we were to do this and fail, then vaccination would be set back decades?
I don't know if it will be decades. We have to continue this discussion for sure, but it would be a big challenge to our discussion here. Yes, yeah. If we fail in the beginning to demonstrate people that we can protect the birds by vaccination, and we get the first, I mean, for field experience, it would be great if these birds survive and all the others die on the farm, but it's also not what you want to see, huh?
And I'm going to do a real quick reminder here. Remember all that they're talking about, because that's going to be the basis of the things that you discussed during the breakout. So you'll have your chance to go through all of this. Some of the audience has already gone through this.
I just want to add, you got to start somewhere. And yes, you know, taking some kind of preemptive action, which is depopulating the entire farm, is an option, but that's not the option that the gentleman sitting there would like to have. So the least you can attempt is to try to vaccinate the best possible way whoever's coming into this humongous complex, which I disagree with. Industry should not build these complexes anymore, but that's secondary. But if you can prepare the pullets in the best possible way and preserve your biosecurity realistically, but in the best possible way, maybe you stand a chance. And maybe if at some point the complex gets infected, well, then you'll have to do away with the birds. But, you know, one thing we did not discuss, what to do with that 18-week-old flock. Well, maybe you can compromise and not use that flock or send it to somebody someplace else. And what do you do with the eggs that you're not producing in that particular house? You always have the ability to extend the lifespan of whoever is existing in that flock. So there are things that you can do. The huge question here is, do I preemptively eliminate the entire farm because it's at incredibly high risk, or do I make an attempt by vaccinating? And yes, Matthias, I fully agree with the fact that if you do this and it fails, it's going to be a huge disappointment and an argument for people that are against vaccination. But again, I do believe that you have to start somewhere.
I agree.
Maybe in a smaller place. Maybe in a smaller place.
So
I don't want to take over, but this brings up a very, very important issue here. The reason why we're so struggling so much with this is because of the tools that we currently have. What do we have? We have recombinant vaccines that can be given at the hatchery or by wing web application if you have the money to spend, 20 cents a bird to pick them up. And the other tool that we have is kill vaccines, which we do know the limitations and the advantages, but they can be given by injection at the same time when you vaccinate the birds for everything else. But we are lacking a delivery system for mass application in production. Otherwise, we wouldn't be discussing this much. You would go in and you could vaccinate by mass application all the rest of the birds in the farm. You'd have a much better chance. So that's something that I think needs to be discussed by everybody.
I want to go back to this picture. Where would the people come from to vaccinate the entire farm? What are they currently doing?
Well, this was my question I put on my paper in the beginning, but then you told me these people are anyhow in the farm to do the vaccinations because you don't have resources outside.
So that's true on the pullet farm, but not for the lay farm. So the 5 million bird lay farm, if we were to vaccinate those chickens, we would have to mobilize them from everywhere around. Where do you find cheap labor who can vaccinate poultry and handle them? Exactly.
Carol, while you're passing the microphone, we have two questions from the audience that are clarification questions. So just to make sure. So the first one is, can you clarify, is this lay farm positive for HPAI already, or is
No.
it just in the zone?
No, it's in the control area.
It's in the control area, not positive. The second, on this case study, can we include a farm premises within the zone that is exporting? So are any of these operations exporting?
They were before the outbreak.
Okay, so they were before the outbreak.
Now,
and
because
once,
of regionalization, that
Yes.
export has stopped.
perfect. Okay, thank you.
Okay. How fun was that? Okay, so turkeys in the brood stages. Oh, yeah, Dr. Clifford.
I got him.
Oh, come on.
It was what? It was a turkey farm?
Oh, come on.
Okay,
Even the
all right.
turkeys.
But it's in the center of that 10-kilometer zone. And where's the layer farm at? Okay.
And the pullet farm.
You can
Okay,
get
so
a,
our index case for that control area was a turkey grow out facility right at the center of that circle. And then for the purpose of this scenario, the layer farms in the northeast corner of that 10-kilometer. Let's say it's 7 kilometers away.
Okay, so now we're gonna look at the turkeys. Turkeys in the brood stages destined for the control area will be vaccinated ASAP. After the first round, turkeys will be vaccinated in the hatchery and in the brooder stage at least 2 weeks before they're moved to grow out. Since the control area will likely be released in about 9 weeks, realistically, vaccination would only occur in the hatchery in this scenario. Oops. Thank God. Okay, so now here's the assumptions we've made. I told you we already got past trade. Here's some other things. Vaccination will be voluntary, but tied to indemnification. So if you want to place birds in the control area and they're vaccinated, you will, you explain.
So the rule would be that if you want to place birds within the control area, so move birds into an empty farm or where there are existing birds, I hope no one's doing that. But in order to be fully eligible for indemnity, if that flock later becomes infected, we would say that you need to have that flock vaccinated. And in addition, currently those farms, destination farms, are required to have a biosecurity compliance audit so that we know they're doing something to help prevent the virus from coming into the farm.
Okay, vaccines will be purchased by the government and the vaccination will be paid for by the industry. Monitoring for immunization will be done by the industry and paid for by the industry. Serology. Virological monitoring for infection will be the responsibility of regulatory agencies and paid for or subsidized by the government. This is very close to the NPIP program. Okay.
Carol, again, a couple real quick clarifying questions. First of all, in the layer operation, you've described the early vaccination program. Are there boosters planned as that layout continues?
It depends on the how long the control area is in place. So that's a question we're going to throw out to you guys to discuss. In our scenario, we decided that the release of the control area was the trigger to end vaccination. We all agree, right?
Sorry, I don't agree. We have those airplanes flying in every time and they drop their bombs and they keep dropping their bombs. So I don't think that this area will be free from avian influenza.
You see how hard it was to work with this group? Okay. All right. Virological monitoring for infection done by the government and paid for by the government. All right. Vaccines will be well matched to challenge strains and updated with annual or as needed periodicity. Okay, that's an assumption for this scenario. So you got good vaccines. Vaccine strategies will target farm to farm spread because we don't know enough to preemptively vaccinate to prevent the next wild bird introduction. So in other words, we would be going for the areas that are nearby the infected farm. We're not going for distant locations because we don't know where wild birds would go. We agree? Okay, I hate to ask. All right. Acceptance or adoption of vaccination will occur among U.S. commodities if trading partners will accept exports. Okay. Oh my God.
You have easy talking. Because regionalization could be an item, and but your country is so much bigger than ours.
Half the size. Right.
Can I interrupt just a
Yes,
second
please.
with a question? So you mentioned the vaccines will be well matched. Did the committee talk about the types of vaccines that they would be using in this
Yeah.
regard?
We'll be there in a second.
You will. Okay, perfect. Thank you.
Okay. Consumers will accept product from vaccinated animals because the industry will develop coordinated messaging, and we won't do this. Okay, as American industries have been known to do in the past. All right. Avian influenza will not be used as a marketing tool. Okay. So yes, we were Pollyannas on this one. All right. You can only use vaccines licensed in the US. Okay. So these are the vaccines that are licensed in the US. Yeah. All delivered by injection. We recognize the need for mass vaccination delivery system. If we were realistically going to take on the big populations in the control area, we would definitely need a mass vaccination delivery system.
So just a clarifying point, no AI vaccine is licensed for use in the United States right now, so.
But these would be the ones that would be. Assumption. We recognize the need for fast tracking of vaccine licensure, but we did not assume it would happen in this scenario.
So another big assumption we're making is that we've got a highly effective vaccine, and this results in virtually eliminating infection rate and causing no clinical signs. So we'll have a difficult time conducting surveillance on these vaccinated flocks to prove that the virus isn't there, because currently our surveillance program is targeted sampling based on sampling sick and dead birds, which works very well when you have a completely susceptible population and reduces our surveillance costs. We currently would collect 2 pools of from dead birds from each house for our surveillance system. We'd no longer be able to do that in a vaccinated population because we would not see birds dying from avian influenza. So I asked our team, our surveillance design team, to estimate for me the costs of our surveillance if we had to switch to sampling healthy birds. And in order to achieve the same probability of detection in a vaccinated flock, we would need to sample, instead of 2 pools per barn, we would be collecting 35 pools per barn. And so if you can, let's just say that the cost just for the testing, not the labor to swab the birds, but just for the testing currently would be about $100 per barn. And under that 35 tube, we're talking about $1,700, per barn. And so that would increase our surveillance costs by at least 35-fold. So we can't do that.
But it's less than the $16 billion depopulation.
Million.
I'm an academic. We don't.
Yeah, and that doesn't include the labor from the producers collect the samples, they collect the swabs and submit them to the laboratory. So that's just a fraction of the cost for that surveillance. So that's not feasible. We need to shift to a different type of sampling strategy for these vaccinated flocks. And so we spent some time working on that. We won't be able to use our routine serology to determine whether these flocks are infected, whether the vaccinated flocks are infected. And we'll need to rely on PCR tests. So we need to find a way to manage that surveillance. we've got to use a lower design prevalence for our vaccinated flocks because we would expect that if it were present in that flock, it would be at a very, very low level. And then we could maintain our surveillance strategy in our unvaccinated populations, or perhaps even focus on those unvaccinated populations in order to demonstrate freedom in the area. So what my team tells me, of people much smarter than me, is that we need to go for a 1% design prevalence in vaccinated flocks at a 95% confidence level. So that's pretty rigorous. And as I mentioned, this increases our cost for surveillance if we have to pick up healthy birds and swab them by 35 times at least. So some strategies that we could consider to increase our probability of detection in these vaccinated flocks and reduce our costs. I propose that we vaccinate 90% of the placements at the hatchery. So instead of vaccinating them all, we'll vaccinate 90%, producing 10% unvaccinated sentinels.
I'm sorry, but in your example, that means that you keep 500,000 birds unvaccinated, right?
Yeah.
Okay, it's well.
Yes, but we don't agree.
I don't agree either, and I'm in North America. But we're assuming here an almost sterilizing vaccination, and we don't know that that's going to be the case under field conditions. We hope that that's going to be the case, but you know, in reality, we're not going to achieve 99% protection, full protection against infection and shedding, such that we cannot no longer monitor our flocks, our vaccinated flocks. Now, realistically, I think we can be a little bit more conservative. With that 1%, my proposal would be, well, maybe calculate your numbers for 5 to 10%, something of that nature, and see how that works. But don't leave 500,000 birds unvaccinated, because that defeats the whole purpose.
I think bucket testing is a really good thing, and you don't need that many birds, but
good thought. So one of the things I was thinking about was the system that was proposed is really looking at an individual bird process. And I think I agree with Guillermo that, and Peter and in the past, is that you're not going to get 100% effective vaccination. So you're going to have built into any vaccination program a number of birds that for some reason, you know, they were missed during vaccination, they didn't amount to immune response. So you're probably going to have birds there that are going to have daily mortality. If you don't have any daily mortality, it's going to be a new occurrence in the poultry industry for anything. So you're going to have daily mortality. But the second thing, I think we kind of had a discussion a little bit about it yesterday, and I believe Tim even brought it up in his talk, is what about instead of thinking about this as a individual bird process of surveillance, why don't we think about this as environmental surveillance? Can we either test the possibility of environmental surveillance, such as could we look at water jugs? Could we look at some other environmental location that would increase your opportunity to get positive samples?
I think that's exactly what we need. We need to get new diagnostic sampling approaches also to not to touch the birds, that we really test the environment and we get many more birds covered by the sampling. And I have one more challenging question, maybe. We discussed a little bit already who is allowed to do diagnostics. We think that the industry should be part of doing diagnostic work, PCR testing, like we do in Germany, for example. What's about the sampling? If you mentioned that the sampling is part of the industry, this could be done by farm people, or this has to be done by authorized veterinarians, or who's doing the sampling then?
Under our current system, producers are trained to collect those samples for us. And then they're submitted to a NALM laboratory, a network of laboratories that are proficiency tested and to conduct the PCR. So they can only go to authorized labs, but the commercial facilities collect their own samples on sick and dead birds.
Because here some of my colleagues may correct me. If you are in a control zone, then the official veterinarians will go out and take samples to prove that birds are negative.
We would not have the capacity to do that.
Yeah, we need trust.
Okay, so that idea was shot down. So what we decided on was to do a combination of approaches. So we'll use our sick and dead sampling, as we always have done, by collecting pools from those sick and dead birds for PCR testing. Then at the same time, we'll also be collecting drinker biofilm sampling on the turkey premises and submitting that for PCR testing, as well as boots and gloves. So you can describe the process for collecting the samples from the boots and gloves. And then we'll do this every 5 to 7 days for the risk period, which is currently the interval that we use within our control area. You want to describe that boot and glove procedure? It's kind of fun.
Sure. So drinker biofilms first, the virus sort of sticks up in the biofilm in turkey, and turkeys have an open watering system. So the virus sort of sticks up in there, and the signal lasts about 2.5 weeks. So it persists beyond what the time period that the birds are actually shedding, and it'll be present in that biofilm. And so we have them collected. They put the swabs into a Whirl-Pack bag, and in the lab, you just add BHI media, squish it up, take out the liquid, and do the same thing for so the samplers, while they're out there collecting those biofilms, they'd be wearing boot socks. Every turkey producer has boot socks because you use that to sample for salmonella. So they would collect those boot socks also into a Whirl-Pack, do the same thing, and test by PCR. You can do virus isolation, but PCR, and then the same thing with the gloves. Just throw them into a bag and squish them up.
So
Unfortunately, both of those environmental sampling tests are unvalidated procedures. So we're going to need to have some folks do the research on that to validate those procedures. Dead birds. I'll describe the dead bird sampling.
okay.
So you have your dead birds, daily mortality from your house. You take your birds and divide them into 2 piles. You sample up to 11 birds in one pile and put it in one tube. That's one pool.
Okay.
You sample up to 11 birds in the other pile of dead birds and put that in the tube. And that's your second pool. So we do 2 pools per house on every house on the farm.
Okay, if I may give in the question from the remote stand. So I had some problems, and now one hand gave me the micro. There was a question that do we know that dead birds targeted sampling doesn't work in vaccinated flocks? Assume 80% vaccine take on whatever vaccine you're given, then on top of that protection won't be truly protective. Then if immunocompromised with some other issues, IBD or whatever, birds with weakened immune system may be more susceptible and thus infected. And this is Ben Weilemann who asked, he just really questioned if dead birds bias sampling isn't actually an acceptable method in large populations. I hope I gave it the right way.
Do we know the answer in the moment?
Not yet, but the surveillance design group is working on that. Right now, the design prevalence for that dead bird sampling is assumed to be 40%. So we assume in our unvaccinated flocks that the prevalence of AI in those dead birds is 40%. So the question is, in a vaccinated flock, what is the proportion of those birds that you would expect to be infected with AI, and what prevalence can we detect with our sampling scheme? And that question hasn't been determined yet. So I'd like suggestions. Do you think it's 10% of the dead birds in a vaccinated house will be infected and vaccinated house would be, the cause of death would be AI or other causes? What do you think the prevalence is?
I'd like to make one comment while they're thinking about that. You'll notice that we're involving the questions off of the computer, but we're not really soliciting questions out of the group here. That's because you guys are going to get a chance to talk about it during your breakouts. The people online will not be able to participate in those breakouts. So I'm sorry if people have comments they'd like to make. We're going to leave that to the breakout for the people in the auditorium. Sorry.
Okay, I don't know the answer, but we will get a chance, as David mentioned, with a farm of 5 million birds, we will have some mortality per day, I think.
Okay, so I think I just want to answer this question. Hi, Ben. I think by doing the biofilm and the dead birds, we're sort of covering our bases. So we know that with 2.3.4.4b, the biofilm sampling really doesn't work. The mortality happens so fast, but we know that in an infected flock, that the mortality would be the place to sample. But we don't know what, as Julie's saying, we don't know what percentage of the normal mortality or the mortality that occurs in a vaccinated flock would actually be infected. So we think without knowing that, we're actually covering the bases. We're sampling the deads and the lives so that we're capturing those two populations together.
I do have a clarifying question that's going to come from the audience.
Sorry, just a quick question. How do you handle the manure on the layer flock? How is it handled? If we want to do environmental sampling, how does the manure get taken off this layer farm? Because if the layer farm is taking manure on a regular basis, one could use that for environmental sampling also.
Yeah, I think we're going to come to that, right?
Yeah, this is Turkey.
Yeah, we're talking about turkeys right now. Yeah, we could go to the pullets.
Okay. Just a sec, we're about 5 minutes to go. So if we're going to layer farms, we need to be moving. So, but a great question.
In a control.
How, okay, we are composting the manure. It's not removed off the farm, it's composted on site. On the ground. No, all of the manure is removed from the buildings and it is incorporated into compost piles that are built on the farm.
Okay, good. Okay, I'm sorry to interrupt, but we need to keep going.
Okay. All right, so our surveillance strategy for our pullets on the pullet farm, vaccinated pullet farm, we're going to strike number 1. That was, we've heard resounding, not lack of support for that idea. And we'll do the boot and glove sampling and dead bird sampling. And we'll do that every 5 to 7 days for the risk period. And then I think we've got another one. Oh, okay, we're on our questions now. You want to take that? Okay, so these are the questions that we'd like you to consider in your breakouts. The trigger for starting to vaccinate. This one, obviously this outbreak is expanding. So at what point in that expanding outbreak would you let a farm vaccinate? You're starting to get requests from farms to vaccinate. And at what point do you say go? On the surveillance, will you accept our innovative approach and which parts of it, or how would you change it? And in this case, is there a need for a vaccine bank and what should it have? What would you stock it with? If there is no vaccine bank, how do you propose to get started? How will you obtain and distribute your vaccine? A few more questions. So we ended vaccination when the infected premises were cleared out, the control areas were released, and 2 incubation periods had passed. So control area release plus 28 days. Do you agree? with our decision to end vaccination at that point? Or what trigger would you use to stop vaccinating? And we obviously had different viewpoints on this, and I'm sure you will too. So what pitfalls did you see that we didn't talk about? And how do these other perspectives impact how you might approach the scenario? So let's just pretend you had to do this tomorrow, like I may have to do. And what would you do?
Okay, so you can see this panel was a little different than yesterday's, which is great. Gives you a little bit of diversity. But also there are some proposed questions to be discussed during the breakout. So what I would next like to do is ask if there's any clarifying questions from the group in here specific to what you're going to speak about in the breakout. Do these questions all make sense to you? Can you recall them once you get up in your individual areas? Do you need any clarifying questions? Okay, great. So we'll go ahead now and we'll put up the slide that shows the breakout, just to remind everybody where they go. And I would also like to throw something in because I happen to have the microphone. There's a lot of discussion about environmental sampling. And I think there would be a lot of value for the breakout groups to at least come back and share experiences and maybe ways to think about the environmental sampling. When I listen to this, what I hear is that it's currently based on individual birds because that's what we have the best understanding for the statistics. But there needs to be more data to understand how you would apply the use of environmental sampling. What's the probability of recovering something in an environmental sample versus an individual bird? So you guys are a lot smarter than me. And just, I'd like to at least have some thoughts on that, or at least challenge Carol while you're drinking coffee to think about that, because when I hear the numbers and I think of the capacity of testing and the resources available to do this, individual bird testing seems like it's going to be challenging in a vaccinated flock. So I think there could be some value in talking about that. And if you think I'm crazy, ignore it. Okay. It's time to start this afternoon's session. Hopefully everybody enjoyed their lunch and the presenters are all prepared for their summary. I think we have a little bit of feedback maybe, but is there another handheld mic somewhere maybe that is? Okay, as long as it's not up against anything electronic, that's great. Thank you.
I'm going to turn.
Okay, so let's start with our presentations. I'd like to go opposite direction this time. So group 5 that were up in the mezzanine, could the presenter please come forward?
Thank you very much. I was voluntold. I would look away for one moment and suddenly my name was thrown in the air and that was it. I was done. So hello, everybody. We had a lot of discussion. I think we ended up with more questions than we did answers, I'm afraid.
Excuse me 1 second. Could you come up here, please? They said you were so photogenic, they wanted to get you on camera. I didn't say that, they did.
I hate being up behind these things. It's nicer being down there part of the crowd.
Yeah, exactly.
This is really high though. So yeah, we had more questions and answers really. And there's some reasons behind that. One of them is the scenario that we were given is something that was looking at a problem 5 years ago when this disease was very different. So we kept stumbling over the problem that what we were talking about didn't feel to us what you would necessarily be doing now, because this is a constant threat now rather than something that's going to turn up once every 5 years. So we had a little bit of a problem with that, and we kept getting dragged back to think about the scenario rather than necessarily the problems. We also thought that scale is quite a problem, and that came up just here when, you know, the size of the Netherlands is, yeah, half the size of the state, let alone the country. And then that leads to a lot of challenges in the US when you're trying to deal with things at such a big level. So that highlights that you can't just take something from one country and transfer it somewhere else and just drop it in and go, hey, great, we've got a solution for everything. Every country needs to be different and looked at in a different way. So we really highlighted what the difficulty of taking an emergency approach is when you've got a disease that has changed. So we're dealing with this short-term scenario when we've got a long-term problem. And I think that's really important that we look at that risk continues. And that comes into that factor of when do you stop vaccinating as much as anything. We did talk about ring vaccination rather than the approach that was being done, because it felt to us that the approach we saw was very focused on how do we save one individual farm. There's one place that's the really important one, and everything else to a degree was sacrificed for that one place that was really important. And there didn't seem to really be a focus of how you're trying to control this for the country or even the state. So it's, hey, we've got one problem here and that's what we're trying to fix. And the gut feeling we had was there was still a lot of risk involved in this approach. If anything, it ran a high risk of devaluing the vaccination. I think the vaccine was being put into a really difficult position. Vaccines are not the only solution, and I say that as a vaccine manufacturer. They are a tool. They're part of a whole set of things that you have to use. And when they're put in difficult situations, then vaccination programs fail. It's not the vaccine failing, it's the program that fails. And that looked like that was a risk that could occur here, where you're ending up with not very many birds being vaccinated compared to how much challenge could be around them. And then if that breaks down, everyone blames the vaccine, and then you're not going to convince anyone to vaccinate again in the future. And, you know, we saw a little bit of that with COVID, where a few deaths which probably weren't related to the vaccine, but everyone jumps on and goes, oh, well, we can't use the vaccine, and you end up with a big anti-vax movement. So we felt that that was quite important. And it also seemed it wasn't necessarily a very level playing field. So you had a situation where it was up to individual people whether they would vaccinate or not and what they were going to do. There was an area of flexibility. But what that opens the door to is a couple of rogue actors who decide they're not going to do anything. They get the benefits of everyone who has done things, while they also put everyone else at much bigger risk. So we were a bit concerned about that approach of just going, we'll trust people to be good and behave well. I will say there are times where that process has worked very well. If we look at Blue Tongue in the UK, which I was part of, that was a trust-based thing and it worked very well. We put a lot of pressure on farmers to vaccinate, and their colleagues did, and they did it. But it does leave a gap that is worrying. We looked at the surveillance, and I think the real issue with the surveillance is you can look at the statistics and say, well, we need to do this number of animals, this number of birds, or we need to do this and we need to do that. But the reality is what resources you have available, and that wasn't discussed, but that really comes down to sheer manpower. You don't want to be bringing a whole heap of extra people in or anything. So how are you going to do all that testing? So you need to start focusing your testing really as a risk-based approach. And the risk is actually outside the vaccinated animals more than the vaccinated animals. So that's part of where you need to be looking at. Wanted to expand that surveillance to more of the farms in the whole area, so we could try and see what's going on in those. And we really felt that the environmental testing is essential for the future, that we find the ways that we manage to do that, because you just don't have the resources to do anything else. Every dead bird should be tested to see whether it's got avian influenza. That's where you start focusing down on some of those issues. And combining different sorts of testing will actually help you because you're more likely to pick things up and get through the gaps if we're using a heap of different testing. We talked about the vaccine bank. That was fairly straightforward. You need a vaccine bank. The sheer number that you're talking about and the vaccines, without a vaccine bank, there's no way any producer is ever going to be able to step up. There's no such thing as surge capacity. Everyone says, oh, great, we can use surge capacity. Vaccine plants run at 90% of their capacity, if not more. And that 10% left is to do with the maintenance that you need to do every year. So there just is not room to suddenly turn around and say, hey, can we have 5 million doses of vaccine? And plants that plan ahead 3 or 4 years in their planning. And then the last bit we talked about was that trigger to stop vaccination. This one is good. I like this answer. When it's no longer needed. One of the comments was, if you stop too early, then you just look silly. So you need to be really sure, and it needs to be risk-based. That was what we went through.
Perfect. Thank you very much. Okay, we're going to get all of the presentations and then we'll start opening up for comments. So if the person presenting for group 4 could come up, please. Up.
Well, first of all, we want to pay our respect to you as a team for this very complex situation and for the solutions you think to have found. We had a very interesting, ongoing, heavy discussions, and what I will be telling you is only a small part of this. So sorry if I left some things out. The discussion started with the emergency vaccination, and we found that an emergency vaccination is an area vaccinating circle around an outbreak. And in this case, the vaccination is done to protect one farm. So we have the impression this is not our interpretation of an emergency vaccination. It's an attempt to protect a farm by introducing vaccinated animals so they cannot introduce AI by the movement of the birds. This is not the traditional interpretation of ring vaccination. And if you want to enter these birds, they should be fully protected. And that was a large discussion in our group. We also concluded that it's impossible to vaccinate the 5 million birds present on the farm. That's not feasible without a mass application possibility. Would you do this? If we discussed that when there is only one outbreak in a free country where the AI is not endemic, we don't see a reason to vaccinate in this case. It's not only we think it's only applicable in this specific area if the virus is spreading faster than the culling capacity. Otherwise, we would not advise to use a vaccination. And we think that a risk assessment is a must to make an inventory of the situation in the area, to protect a single farm if this is worthwhile. If it is a short break, why don't you set all bird movements on hold? Then regarding when to stop the vaccination, if it's spreading in the area, there is no discussion, you should continue. Although in the way we're doing it in the proposal is you would need more than a year to protect your whole farm. And is this worthwhile? Secondly, if the infection is in wild birds and it has become endemic, well, you should start preventive vaccination based on a risk assessment. And also here we see no emergency in only one farm. Then regarding the sampling, we thought that you should sample all vaccinated birds, all vaccinated flocks, sorry. Small difference. We discussed about the 10% sentinel birds in the flock, and we thought it was not wise to have any sentinels in the flock. Regarding testing, we would start testing by taking swabs of the dead and the ill birds, the current testing method. but we would strongly advise to take at the same time environmental samples, like the Germans do, for instance, and to get a validated testing and sampling method for these environmental samples. And I think then I covered, oh no, then there is another topic, sorry. Then we have the frequency of testing. We were talking about the costs and the implications this has. Sorry, I can't read my notes anymore. It was so hurry. We were discussing about the R and what's the definition of the R. If the R is below 1, the virus is not spreading in a flock. So the frequency can, in our case, be less intensive. If you have no information about the vaccine, we think that a weekly sampling should be something to start with in the current situation. Well, it seems most appropriate. That's it.
Perfect. Thank you. Okay, let's see group 3, please.
I think it's only appropriate the former CBO of the US gets to do this, huh? I was there during the 2014-15 outbreak. So was Julie, a lot of other people. But anyway, so this was a lively discussion with our group. I'm not sure we ever came to a conclusion on especially the first issue about wind vaccination. One of the things we did recommend, though, highly is a qualitative risk assessment right away. If there was also lateral spread, we talked about ring vaccination. Some people felt that we should vaccinate in the entire area, some not at all. So we had kind of we never came to a final determination on that. However, I guess because I've got the mic, I think if there's no lateral spread, or if there is lateral spread, I think ring vaccination is appropriate around the 10-kilometer area, not within the 10 kilometers initially. If there is no lateral spread, and depending upon whether you're going to use vaccine long-term, if you are, then you get population after you remove controls, you get the population vaccinated. If not, you don't vaccinate any of them, because I think you're not going to get immunity in that area fast enough in order to really protect the birds, and you're going to have disease spread. On the issue of surveillance, we liked your ideas on surveillance. We do believe environmental sampling is definitely needed, as well as dead bird testing. But whatever we use, we got to have validated from the standpoint of making sure that it's truly scientifically sound. And that includes also, can you use biofilm on nipple waterers? No, you can't. So that would have to be addressed in another way. Maybe dust swabs, maybe even air sampling. There's some techniques I just saw where they can do air sampling in these houses. On the dead birds, we recommended basically, I think, which is the standard you all use, 11 swabs per 50 dead bird pile, 11 swabs per container. And for chickens and turkeys, do oropharyngeal swabs. For ducks, do both cloacal and oropharyngeal. Let's see. For the control zone, as far as incubation periods, we agree that 2 incubation periods is appropriate. However, with regards to vaccination birds, there would be some discussion that needs to be validated with vaccination if it's heavily used in that area. Plus, some people felt that if there was a high wild bird population in that area, that you might need to extend that period of time based upon that. And it's really based upon the level of risk and the density in the area, whether you release after 28 days or not. So I think that's pretty much it.
Okay, great. Thank you very much. Okay, so group 2.
So I'm supposed to tell you what we found out in group 2. At the end, I've got the microphone, but we are quite close together with our opinions. Especially yesterday, we already decided, the whole group was 100% for saying we don't need sentinels in times with PCR. So this, I think, covers quite a lot of what I'm going to go on for. Going to the first question, when to start was the answer was yesterday. So if there is something in this area, you're too late. And with the vaccines we have at the moment, we don't think that you have a chance to go. You need an army to vaccinate a flock like that. But I think the best answer from this group would be to say we need more vaccines. We need more knowledge from vaccines. And from Mexico, for example, he told us that they have a vaccine on an ND base with H5, but with good ND titers in the flock, you have no chance to spray it. But maybe there are other opportunities. And if we get to the message that we will start to vaccinate, then there might be a big market for the industry, and we will make bigger efforts on finding other strategies for seeing how to prevent, or if it's an emergency vaccination or preventive vaccination. We think that we have to go to the preventive vaccination, and the hatchery is the best place to do it. But also there we need more information. How long do we need a booster, or what are we going to do? For your flock, actually, we think you don't have a chance to go in with a vaccination. Maybe you have to think about selling the pullets, not to bring them in, and mold the ones you have for times like this, because every time you move birds into such a big system, it's very critical. So we see the highest risk in the personal and the truckways if you open the system to bring something inside. And even if you think about just vaccinating this one big unit, but you have a high pressure in the surrounding by not vaccinating the others, how you're thinking about at the moment, we think that the risk for it to jump over will be much higher in that place. Yes, going to the surveillance, I think we need more information. For Germany, we do the bucket testing and several more things. If you discuss about your genome type, or if there are differences and you don't find it, actually by doing a huge amount of testings, if you're very close, maybe you test them daily, then you're in front of the business actually, but no one can afford it. But we need new studies on more information, and I think we have to do it very quick, or the group thinks we have to do it very quick, because we need it to make sure for the WTO, for example, that they are clear. We know what we do, and we have the right instruments to be sure that the vaccinated flocks were on a good protection side from we know what's inside, virus or not. So we need to validate environmental swabs, drinkers or something else, what's possible. But if we do it with a farm personnel with no cross contacts in an easy system, I think this will bring down the costs and make us quite close to the flocks and let us see what's happening. The other thing is that we need more information for the PCR, because we think it's the best way. Serology is always too late. And for the PCR, we have to know how big the pools can be, and all these information should be also validated. Yes, and for the dead birds, we know from Germany that if you sample the good dead birds, no cannibalism or something like that, then we think we have a 100% chance to find the virus. But we also need to validate that probably on a huger amount. Yes, and the third bigger point is when to end the vaccination. As soon as we don't find any virus in the wild birds, and also, of course, in backyard and farming. This is our opinion. Thank you.
Okay, thank you. So number one, the last group. Up. Carol said we saved the best for last.
I'm afraid not. Okay, so I'll have the difficult task to do justice to group one and the lively discussions we had there. And just to sort of come to the conclusion first, we very much agree with what has already been said by the other groups. So we were sort of discussing in the same direction. First, about what triggers an outbreak and whether we would vaccinate that farm, and we concluded no, probably not. One reason being is the time of the vaccination, the timing as well. So it takes time until actually protection would be there. And we were sort of asking what's the point if only this particular farm would vaccinate and not the other. So we were then also going more to the conclusion or the discussion, it would be better to go for a larger area to vaccinate, including ring vaccination. So definitely no voluntary vaccination because protection of the whole flock, of the national flock, has to be the goal if vaccination is applied. And actually, right with this first question, it started the fundamental discussions about the vaccination to kill or vaccination to live, because that also would have implications for the decision. I'll come back to that later. Good. Surveillance. We very much welcome the environmental sampling, like all the other groups have done as well. And we also included or briefly discussed the option of air sampling, but also sort of recognize that with the methods currently available and assuming that we have a rather low within herd prevalence that we would want to detect, probably the sensitivity of these approaches is not quite there yet. Bucket sampling, absolutely, we completely agree with that. And we also thought the socks would be good for the turkey farm. Pooled samples of dead birds, also welcome, we agree on that. Need for the vaccine bank, yes. But we also sort of thought we probably would need different types of vaccine in such a vaccine bank, because we would have to see what is the purpose of the vaccination, preventative emergency vaccines for short and long living birds, etc. So different types would be needed, which antigens to consider, probably likely we would need 1 to 2 strains, and that would have to be risk-based. And if we are going to, if we're going to go for a vaccination to live, we also have to consider a boost that would have to be applied. So that also needs to be considered when setting up the vaccine bank. And we would recommend replicant vaccines for such a vaccine bank because they can be quickly produced, in high number of doses. And if there is no vaccine bank, what would we do? Well, we said we would pick up the phone and hope for the best. So basically, bottom line, contingency planning is required for such situations. What triggers end of vaccination? Here we need data to be able to come up with the decision. So we need surveillance data to actually be able to say now is okay to stop vaccination. So it depends on the wider context. And even if we stop vaccination, surveillance on vaccinated birds would have to continue. And sort of at the conclusion, what else to consider? We thought probably what wasn't sort of discussed much was the public view on these measures and on the situation, sort of what is the social dimension in this whole story. And we also sort of came to the conclusions about this concept of emergency vaccination. Is that maybe a flawed concept that may have worked very well and that nobody would questions for situations like foot and mouth disease, but maybe it is not transposable to AI? Is that maybe something we need to reconsider overall? So I think it's good to have now a question at the end of the presentation. Thank you very much.
Okay, thank you to all of the presenters. Despite the fact that you were nominated, you all tackled that responsibility with enthusiasm and good presentation. So thank you. So now we're going to ask the panel if they heard anything that stimulated thought, or if they wanted to make comments on any of the comments that were made. And we'll go through that and see how that works. And then once we've had some of their feedback, we're going to open it up for questions. Okay, so first, anyone have a comment they want to make?
I'd like to remind everybody, I don't know, I think it's the second Star Trek movie where they talk about how Captain Kirk was the only person to solve the unsolvable thing at the Star Academy. And it's because he reprogrammed the computer because the question was impossible to answer. So that's what our groups here did. They reprogrammed the computer to ask questions that actually can be answered. And I think that's the way we felt on this panel. Faced with a mega farm and the idea of emergency vaccination, it can't happen. You have to have started a long time ago, and the planning and has to be there. There's not enough vaccine, available to do this one control area at this time. So there has to be, and I think you guys all came to that conclusion. So thank you for reprogramming the computer.
How about the turkeys? Yeah, just wanted to remind everybody that we did vaccinate the turkeys. We had about 50 flocks in there, and anytime we were replacing a flock and moving them in, those baby turkeys would be vaccinated. So we weren't the objective wasn't simply to protect that big layer flock, although that's a big incentive. But in this actual situation, a true life story, in a control area where the disease is raging, it becomes impossible to get a turkey to market. So we are depopulating every single flock that's placed in that controlled area so that company cannot get a paycheck. The government's paying for all of those birds that become infected. So we're trying to preserve the ability of that area to raise poultry in this case. So that's one thing to consider. We were trying to protect those turkeys. And the other comment, this might have helped you. The risk assessment, the case control study that we did showed that a farm that was in a control area is 10 times more likely to become infected than a farm outside of the control area. So that might have been helpful information as you're doing your deliberations about the risk to flocks within that area.
Okay, good. Sorry, please.
Well, it's it's difficult to. It would be a summary of the most important things. I think emergency vaccination for me is unrealistic. Okay, for us in Europe, anyhow, 5 million birds is unrealistic in one place. But even in smaller populations, we know if there's an outbreak, then we need the people for depopulation and not for vaccination if you want to stop it, because there's no chance to stop it by vaccination in an emergency situation. We have to do it preventive. And other things like diagnostics, collaboration between industry and state officials, it's absolutely needed. And okay, it's just a summary.
I'd also like to say thank you to everyone who agreed with our surveillance plan.
Well, I don't know, but in my view, considering the current situation where you have such a widespread infection, it really is not realistic to think that you can go in and vaccinate yourself out of an outbreak. You probably have to compromise a lot and lose a lot by depopulating, and then strategizing to start vaccinating whatever is going to go to areas where you are much more likely to succeed. Because if you think that you can go into an area so densely populated like that that is raging with virus infection. And vaccinating birds, I think you might succeed in some cases, but it's going to be very difficult in the end. Just kind of personal feeling.
I don't agree. Sorry. But
We're all surprised.
I'm glad that you all had the same discussions that we had. And I think we had an impossible task here. But it was nice to work together and to have these different views and to discuss them. So, and I think a lot of points that were mentioned here by the groups are all correct. But that leaves us in the end with a question, actually, and a question mark. And I would really like to see a sort of a vote about that. Me personally, I'm very much in favor of vaccination, and I would like to get that going on. But I would also like to know from the audience, from everyone, what you think about vaccination. If I put the general question, are we going to vaccinate?
Say preemptive plan.
Preemptive or not? That's the question. So is there a good way to say, raise hands, or can we use the system?
No, I don't think we can use the system, but I think if you say raise hands, we'll get a feel for it. We'll get an order of magnitude.
Okay.
Well, I won't be able to validate the number, but we'll have an order of magnitude.
Well, then I'm going to pop the question. Who would be in favor of a preemptive vaccination? Please.
Yeah, there's so many questions.
Well, it's preventive. Yeah, preventive. Okay.
Okay.
And maybe to get this question that you popped up, so I'm going also to ask the question: who is not in favor? Okay. Yes. That's one. That's good.
Yeah. So it'd be good to. Here, I'll take mine. You can keep yours.
Yeah.
It's because of trade. Because if all these people out here in all these countries will accept vaccination, then there may be a different answer. But right now, they won't. And in fact, the US regulations don't accept vaccination. So we're going to lose 20% of our market. 20% of the US industry exports goes out of the country, 20%. It's $4.5 billion that we lose. In addition, in the US, Carol's right, the broiler industry is against vaccination. The primary breeders are against vaccination. The turkey industry, the NTF, National Turkey Federation, is for vaccination as long as they don't lose their markets. So the only ones that really support total vaccination is the egg industry, and they ship 3% of their product out of the US. That's where we are. And we've got agreements with 80 countries.
Yeah. And that's the answer that I get here. It's the trade, stupid. So, but what I really liked about here is that you are all saying, yes, we would like vaccination. And if we go and work into that direction, and we get the EU OIE, and we get the European Community, and all the things that are happening there, they are great. So if we all agree and we bring that message to our countries, then maybe we should start somewhere. And then I like the remark from yesterday that maybe a country should try it out, and then without the other countries punishing this one country. But okay.
Perfect. Thank you. Okay, so we have a question from the computer.
Yeah.
AI question.
This is
Challenging.
Okay, so I'm going to read the question. Several groups said a risk assessment should be done. What does this mean? More specifically, what does the risk assessment need to show in order to trigger the use of vaccination? And more specifically than that, if vaccination is deemed appropriate from the risk management perspective, what level trigger is acceptable to other countries to say vaccination in your situation is okay and no trade impacts other than what are already existing in the control zone or the region, et cetera. And lastly, rigorous risk assessment in our experience equals years and thusly not particularly responsive. By then, we may have been too late. Perhaps this is the real outcome of this meeting, is a risk assessment, things needed to be filled out, and levels that trigger use of vaccination. So, yeah, and then there's a follow-up one which says change the trade regulation. It's just very clear to me whether it is animal welfare, sustainability, and feeding the world's population specific to the poor. So going to the first one first, please, and the risk assessment. Can somebody speak more to the risk assessment, recognizing that I'm bringing you the microphone? Thank you.
Yeah, I would totally agree. Qualitative risk assessment is going to take us 2 years, so it's not really helpful in this situation. And then what are you going to do with the results? Do we have those triggers? Do we have those, what is the level that we're looking for in that risk assessment to say, go?
Proposal was, correct me if I'm wrong, but there was a proposal in there to do, to proactively do risk assessment. Was that right?
Yeah, I think you can read that into it. And then the question is, can you proactively do a risk assessment?
Could you proactively do a risk assessment around the different types of scenarios that might lead you to a trigger?
Yeah, it sounds like an extension of the secure poultry supply plans, doesn't it?
Chapter 12.
Another grant application.
Yes. I think that's.
No, take it back and please.
I think that's an excellent suggestion, and it's not going to be a perfect solution. There's going to always be new and different situations that come up that you wouldn't have covered, but I think that is something that we struggled with mightily up here. When do you pull the trigger, and when do you pull the trigger for different commodities? We clearly cannot steer the massive ship that is 5 million birds. We can't. Right? So you have to have some sort of planning and preemptiveness around it. So I think that, I think proactive risk assessment, I think is an excellent suggestion.
Okay. So any other questions on the, not yet. Okay. So if there's no other comments from the panel and there's no other comments specific, oh, sorry, we do have a comment here.
Thank you, Peter. I couldn't vote. I actually abstained. Okay, because I think the problem is the trade question hasn't been answered. So I'd ask the gentleman who stood up and actually voted against it. If the trade was sorted, would you be in favour of vaccination? Because I think certainly some of my UK colleagues would have probably would have voted another way or probably abstained, because it comes down ultimately to trade. We have to sort out the trade issues. So the question to you is, would you be in favour of vaccination if trade was sorted?
Well, I'm not the CVO anymore. So
you voted.
Let me just tell you this. I think I'm not going to, this is trying to put me on the spot, which I'm consulting for the U.S. poultry industry as a whole. So I think there's value in vaccination. Okay, I do. And I think it should be used when it's of value, and this situation what we have and that we're in now, is presented itself where I think there's value in having vaccine in use. The problem we face, if trade was totally resolved, I think, you know, the US might look at it differently, but I would have to go to USDA and ask them what their issue is, because in the US, APHIS Veterinary Services has to approve the use of the vaccine. And then also in the US, every state veterinarian, that's head, the chief veterinarian for that state has to approve the use of vaccine in every single state.
So if the federal government approves vaccination, how many do you think of the state veterinarians are actually going to back off and say no? How many of them are going to do that?
I can address that. The state veterinarians are asking for it.
Exactly.
They're asking us to vaccinate. And so if trade were not a barrier, we would probably be doing it today.
So, but we've got 80 countries to, you know. Well, you know, for the EU, look, I mean, yeah, all he has to do in the EU is for EU Commission to say you're all going to accept it.
Hold on a second. You want to make more comment?
Just a comment saying when you talk about trade, you always talk about the others, but trade is ourselves first. And when you talk about the global trade of genetic of animals, of poultry, how many countries do you have for that? And all of them are here in the room. So it's up to us to agree, not to countries that are 5% or 2% of our exports.
Okay, thank you. Comment.
My name is Birte Steenberg, and I'm the Secretary General of AVEC, the European Poultry Meat Association. So I'm sort of the same for Europe as John now is for the US. And I can say on behalf of our members that they have said no to vaccination, period, until the start of this year. And they still say, well, we need to do something, but if the trade issue is not solved, then we can't go through with it. And I think that what we need here in this group, we have fantastic scientists on vaccination, on veterinary issues. What we really need is somebody that can communicate much better than us, so we get the trust within our trade partners. We discussed it in the group I was in yesterday. We really need the trust of our trading partners, and I don't have the solution for that. I'm a veterinarian. I'm not a communication expert, and communication has become a science in the last years. So I really think we should team up with some people that can help us create the trust between ourselves, but also between the countries that are not here today.
Thank you. I have a question, if I can, and maybe it's not the right question, but there's been a lot of discussion about building trust with trading partners. And it suggests to me that maybe there's not trust or a lack of understanding or whatever. But my question's more basic. What will it take to be able to open those discussions that can facilitate building trust? In other words, what do we need to know and what do we need to share to be able to have conversations to generate those 80 agreements or however many agreements? But what's it going to take? Trade sounds like a barrier, but I'm not sure I understand what the strategy is to overcome it.
So in the US, when we started doing zoning with our trading partners, especially in 2014-15, we went in and explained the entire US program to them. We sat down with them face to face and explained this program, and we still do it today. We go, USAPEC in conjunction with APHIS goes around the world. We go to Dubai. We went there and talked to about 16 Middle Eastern countries. We were just in Africa and Rwanda and talked to 18 African countries to explain this. You know, the CVO from Taiwan is here. I've been personally with her and her staff when I was the CVO, and even now in my current role. That's what it takes. You got to talk to people. Now, what's it going to take with vaccine is for them to trust the system that I've already explained to them and explain how we're going to continue to protect them with the use of vaccine. That's what it's going to take. And for them to accept that.
And I'm not going to completely go away from that, because what I'm curious about is when you go in and you talk about it and you want to convince somebody, I think I heard Julie say it takes science. Okay. Okay. But there must be a willingness to listen when a country comes in and it shares its plan. It says, we do this, we do that, we do this, we do that. I'm just wondering how that becomes adequate for people in this room to convince them, yes, maybe we should try an experiment. Maybe we take a 20% area of the Netherlands, we allow them to vaccinate, and we get an agreement that they can, in fact, continue to export, and we do a big experiment like that. But I'm trying to figure out what it takes to actually convince somebody to do that.
That word adequate drives me crazy. And that's what we have from Waller right now. That country will, you can have freedom with vaccination, provided you have adequate surveillance. Does it go further into describing what adequate surveillance is? What that is to me in my country is very much probably different from what you would consider to be adequate. So can we frame adequate in a more detailed way?
If I may, there are no questions but comments from our remote colleagues. And I tried to give a little bit impression of the conversation. If the trade is the issue, when we have HPAI outbreaks, there will be no product to export. Full stop. And more than anything, for animal welfare, for human beings, how can we still look at this picture to cull or not? Can we imagine this during the COVID to cull humans? We just had the vaccine, and there you are being in the room together thanks to vaccines. And this is a perfect time to experiment with vaccination for 2, 3, 4, 4, and this count lineage is nearly worldwide, or shortly will be. Thus, very little risk of messing this up or ruining a current country status, and so on and so on. So there is a clear option on vaccination.
Yes, I agree. This time. But I was, yesterday I was surprised, but I was happily surprised. I saw the presentations of the OIE, and I saw also the presentation of the EU, and they are changing the rules. I was not so happily surprised that it would take another 2-3 years before EFSA is going to give its opinion, but okay, that's something different. But saying that, if we change these rules in such a way that these bodies, and they are, let's say, well-known good bodies, if they say, okay, this is the way that we can go, if we do it like that, that, and that, that would be maybe a good starting position for everyone to trust that. And then there is a basis of trust. And from that point on, you can go further. And yes, I was also thinking about the Netherlands and doing a test there, but I'm not the one who's going to decide that.
I see somebody in the front row that might be willing to.
Yeah, thank you. Just to come back to this thing about science and the fact that we asked EFSA, I was asked to do a risk assessment. It will take time, of course. The problem if we miss some science, the decisions will not be ours anyway. At some stage, some politicians will say, I can't bear anymore this situation, and you will have to vaccinate it, basta. Who cares about the trade, etc.? How much do you get from trade and how much do I pay? So exactly the same comments that was said on the thing is, so what is wishable, I don't know if it's a good English term, but wishable is to get some
guidance from the scientific from the scientific community that we are, and maybe ask WOA for a little bit beyond, because what we have in the code is very, of course, it's very high level and it's adequate. So it's not really adequate for us, but to ask more, but in a way that it is still adaptable to all the situation and to the risk assessments that we will get from our scientific colleagues. But it's true that at some stage, the decision will not be taken by us.
Yep. You might need to tighten more. You might need to tighten that microphone. Starting to.
So to the person that sent in the message, about vaccine, from a scientific pure point of view, yes, they're absolutely correct. We need to be using vaccine. Unfortunately, we don't live in a world where science just reigns all the time. We live in an economic world as well. And while I come from a farming background and I care about agriculture, I care about American agriculture, I care about people like Chris, who's lost money and can't vaccinate to protect his flocks, as well as turkey industry and everybody else. But unfortunately, where we are today, unless we can get those guarantees, it's not just losing $5 billion in exports or 4.5 or whatever it is. It's more than that, because we're going to be losing close to 20% of our broilers, which means, which means you're putting a lot of people out of work, lots, not just a few farms, lots, 20% of that group. And so all that has an economic effect. In addition, the breeding industry in the US, unless they know they can ship, have indicated to me, if they lose those markets, they're going to have to move some of their facilities out of our country. There's another loss to people that work there in that breeding industry. And so you're talking about billions of dollars, folks. So it's not that easy unless the OIE or somebody, or including ourselves, can get guarantees from this country. There will be no action if you vaccinate.
So one of the purposes of these types of meetings, and thank you for those comments, they're very relevant. One of the purposes of these types of meetings is to come to some sort of an agreement on what's needed and be able to share recommendations on how to go forward. I haven't been involved in these meetings historically, so I may sound naive, but what I think I've heard is that there has to be a certain amount of scientific input relative to a justification for adequate surveillance. And that in my mind ties with the risk assessment, which says, how do you make a decision when you can vaccinate? And that those two pieces are tangible pieces that could help when the industries then have to approach trade partners and regulatory authorities to discuss ways in which preventative vaccine could and could not be used. And I still, I go back to that analogy yesterday of, putting your toe in the water. I'm not sure that we're ready to put the toe in the water necessarily and take a jump and start vaccinating and see what happens. But I'm sort of thinking that we ought to at least make sure there's water in the pool. And it seems to me that there's a couple of things that are being identified that might be helpful. Now, I'll look to people a lot smarter than me, like David and others, to say whether or not this is something that is productive and we should include as outcomes of the meeting, or if it's something that needs to be reconsidered, restructured, or whatever. And I was setting David up because I know him and I see him holding the microphone.
Yeah, you must have read my mind. You said everything I was going to say. Appreciate it. I was going to say that I'm not an epidemiologist, so I'll have to rely on Julie and Ara and other people in the room. But in deliberations on several ad hoc committees, when you start talking about surveillance and you start talking about epidemiological issues, it's exactly like you say. Simplistically, as a pathologist, I would like to say, well, I want A, B, and C, and I want you to say that in the code or in the manual. But you can't do that because you're talking about each situation is different. In the case of poultry, we're talking about the species and their susceptibility. You're talking about the individual virus. and what species it's adapted for. You're talking about what's the poultry density in that area. So there's all these factors that go into deciding what that surveillance is. Can you do targeted, or do you have to do broad general surveillance? So I think we just need to make sure we recognize that World Organization of Animal Health is not bucking their responsibility, because they can't really write a prescriptive surveillance program and put it in the code, because there's so many scientific inputs that have to be taken to do a proper surveillance. And I think I think the challenge for, I think what John is saying is we have to convince those trading partners. And one of the big questions is, how do we convince them that we have a surveillance program that's going to meet that need? And maybe the starting point is, we have a surveillance program for non-vaccinated flocks.
Yep.
We've got to show, does our program for non-vaccinated flocks also adequate for vaccinated flocks? So we have a groundwork that's been laid, but we have to, it's unfortunate, it's going to take a lot of work, because we're going to have to go back to them and say, this is what's done. Now, it could be, a suggestion might be you could get together the key trading partners, and try to come to some agreement scientifically what really is needed. And you could maybe shorten down that individual country to country requirement. You're still going to have a country to country discussion, because that's the way all the trade agreements are written. You can't get around that.
Right. And so that's a good expansion, and thank you for that. I think we have a comment from, there's a question in the back, and then while it's going back, or did somebody up here have a comment?
Thank you. My question is, if it's really black or white with the trading, so do we have to think about other ways to go to the problem? Because if it's some flocks are vaccinated, does really the whole country lose the trading partners? This is one thing maybe. Or the other thing is, let's discuss about regionalization, even if it's hard for the Netherlands, but for others, it might be an opinion or an option. So if you go to the Russian papers at the moment, they have regionalization, or they have companies on their list. So maybe we have to make the problem a little bit smaller by steps in between.
I agree. And we wish to use regionalization with vaccination. The problem is that we do not have a single agreement that allows us to export with vaccination, regionalization or not.
But I think we are discussing vaccination. It's not black and white. We have many options for vaccination. The same is with trade. We put everything in one pot. We, as a primary breeder company, for example, I think the others agree in Europe, I would never vaccinate our breeders, our primary breeders. So we are kind of compartment or region in the country, and we prove that our flocks are free, and we still cannot export because the trade partners will block this. So this is something we also have to explain them. You vaccinate susceptible populations in your country. You have a good surveillance to prove that in case they get infected, you will still stamp them out. And this we have to try to tell the people, and why we do it. And in the first case, we as a primary breeder company, we do not want to export from vaccinated flocks. They are all non-vaccinated, and they are proven to be free. So it's the trade and vaccination. There are different policies and options to do, and we have to. It's the start to discuss this in a big group, and for sure we will not have a conclusion today, but we have to continue.
Rick, go ahead.
Yeah, I just would like to second on David's point regarding the unvaccinated situation, because somehow we also seem to assume here that well, the trade issue with unvaccinated poultry is at zero risk, and it definitely is not, because also, well, in many places the virus is falling from the sky, so also there is a risk, and yeah, you always have a window where you did not make the diagnosis, and you could have it. And it would be good to we mentioned this risk assessment for this case studies, but in general to have these two situations compared for the trade issue. So how is the risk now, and if that is the threshold to be reached, how can you design your surveillance system to have a risk at least that slow. That would be, I think, a scientific way to approach it.
Thank you. I want to second Julie on the issue of zoning. I think that's the official WOA term versus regionalization, zoning and vaccination. And I think really that we need the help of WOA on this, because today we have text in standards about zoning, we have text about vaccination, but they don't match together. So maybe that's something that we should give WOA food for thought. Also, what regards adequate surveillance, I had this discussion with Etienne, I think a while ago, and Etienne, if I remember right, you told me it's all in the manual and there's a chapter, there is no need for further indication. All the surveillance that's needed is here. But is it really? Could it be a topic that we have at the next general session of WOA when avian influenza is going to be discussed? So that's points I wanted to raise. Thank you very much.
Yeah, I don't want to put anyone in the room in an uncomfortable position, but I think we've probably got some people in here that are representatives of trading partners. And I kind of like to get their perspective. Are they more assured, based on what they've heard over the last 2 days, that we can fix this problem? John made the point there's 80. Okay, you can't have a dialogue with 80, but we have to start somewhere. And I'd be keen to know, what is it? We have to build the trust in the surveillance systems, as David said. What is that real stumbling block that's causing the concern? And have we not gone far enough to show we got an evidence to unpick that?
So he didn't want to put anyone on the spot. I will. Is there an, oh.
No, it was okay. I was just, when you were talking about compartments earlier, in the US, our breeding industry, our compartments, I don't know if Highline finished their compartment, haven't finished. So 2 of them are Avian and Cobb. And we only have agreements with 2 countries on them being compartments.
But it is a step in the right direction. It's a step in the right direction as far as moving towards the recommendation. Maybe I see it. Not in Europe. Are there any representatives of trading partners that would feel comfortable to make observations? It's a good group. It's a friendly group. Oh, she didn't hear the question? Okay, who are we speaking to? The woman from the empty? Oh.
Thank you to give me the opportunity. I'm just on behalf of my colleagues from KSA, Saudi Arabia. I'm the chief veterinarian for Amrai, one of the biggest producer in Saudi Arabia. I think we are, our concern in KSA is going to be the trade. But I will make it a simple way. We need to agree that you need to convince us that the surveillance program was you're going to use, and especially the traders. We trade with UK and Netherlands. We're talking about 2 different things, is genetic materials of the parent stock or grandparents. That is a fundamental that you need to make sure that those birds not being vaccinated, but we need some kind of mechanisms. How are you going to make sure those birds arriving to KSA and there is no silent infection? We discussed that a lot, but it could happen. You're vaccinating some population and some not. The second point is more important. We have millions of hatching eggs coming from Europe, especially from Netherlands and Belgium. Now, if Netherlands decided to vaccinate east of Netherlands, northern Netherlands, we need to discuss this now. We cannot discuss this late. We need to open the channels to discuss with the KSA. I think at MEWA level or the Ministry of Agriculture, we need the chief veterinarian from Netherlands and UK to start negotiation and discuss the deal, how that will work. You know, what guarantee you will give us? We are not against that you're going to use vaccine. We want you to survive this crisis because we want as well to survive, but in a way that we work together, you know. And now we need to discuss this, not you are guys discussing between you about the trade and leaving us what we are the biggest partner with you. You know what I mean? Thanks.
Thank you. So in terms of the risk assessment, you not only have to think about the type of bird or the type of operation, but you also need to think about the individual commodity coming out of that that situation. Can AI be shed through the egg of an infected hen?
Yes, a high path avian influenza will be shed in a certain percentage of eggs produced by an infected flock. However, the process of incubation would render what hatches out of that incubator to be harmless and free of AI.
Sounds like a bullet in a risk assessment to me.
If I may add something, here is a very vivid discussion on the remote participants and myself, so I do not want to bring everything, but give you an impression. There is a strong wish to change the current regulations and to allow vaccination and lower the trade barriers. And I think what was said here, open the channels and go to your CVOs and go to the experts in the relevant working groups and say, here we have to do something. This is the only thing we can do at the moment, and we should do it. But this is also the opinion of the experts here giving their comments online.
Okay, we've only got 2 minutes left. So I'd like to capture just final comments. It's been a good discussion. Thank you. But I think that we'll have to wrap up with this final comment and one comment here.
Okay, thank you very much. We were talking about key trading partners, and we are talking about that Mexico, Canada, and United States has maybe the biggest free trade agreement in the world, and Mexico vaccinates. So my question would be, what kind of, how can we build trust in order, because we're talking about trust, in order in free areas, that we have free areas of avian influenza with no vaccination, that what kind of surveillance or compartmentalization we have to do in order we could export to United States. We are importing from the United States. So this is part of the game, no? And I think 2 or 3 we have to go to the swimming pool together to see if it is if it will work. No?
Good comment. Thank you.
Thank you very much. Just to bounce on it and Olivier's talk is about how to build trust. It's true that having standards is a way to build trust, because if you have common, harmonized, standardized, and adopted and commonly adopted way of dealing with the issue, that is much a big step towards trust, common trust. And it's true that in May, one of the technical items will be on avian influenza, HPAI, and maybe vaccination, of course, will be on top of this on this item. So there will be discussions, and there will be the members, as I said, will be demanding or not. I mean, if they demand more guidance, more harmonized guidance, we will do what we can, because of course, as David said, we cannot go to the details, but if we have something, like we said, for other diseases, like for, there is already guidelines for compartmentalization of avian flu on the website of the OIE. So, but let me just tell you one little story about FMD. When we began to say no FMD, we can't bear any more FMD without vaccination, we want to do vaccination, etc. And we built trust about vaccines. It's a nightmare vaccination for FMD. We all know that it's difficult, but we built trust, etc. We had the OIE, the OOR giving status, official status adopted by 182 members that did not, and that will not avoid trading partners to still continue to have problems with this or that problem of resolution of I don't trust your surveillance, I don't trust your vaccination, etc. So watch out. WOHA standards, yes, this is part of the trust. And indeed, we hope that will be a way forward in this direction. But as was said by another colleagues, bilateral discussions and a long-term bilateral discussion about how we deal with the specific topics in the country is something that we will not be able to avoid. And it's already the case with no vaccination, because regionalization on AI is something which is already very difficult.
Right next, please. Thank you.
Yes, thank you very much. No, just to echo or I can support most of what I heard in this discussion, and I just wanted to confirm that indeed the European Union is taking a huge step forward with these new rules that we're currently putting in place. And obviously things take time. I mean, the EFSA opinion will take time. The trade discussions take a lot of time. And so this is not something we will solve in a few weeks or in a few months, but we have to start somewhere. And so this is the first steps that we are taking now at EU level. And we hope that, you know, some people will jump in the water, and then we can start small and then build experience and then enlarge. And I'm sure that the trade issues will also, I mean, can be overcome, but it's something that takes time. Thank you.
Perfect. Thank you very much. And on that note, what I'd like to do is, first of all, thank the panel once more. This was a tough one and they did a good job. And then next, I have the privilege to tell you that you get to take a break.